what we're going to be looking at, as you can see on our second slide on the first page, is the central nervous system. We're talking specifically about the brain and the spinal cord. What we're going to be doing after that is really just describing some of the function. This is all we're really looking at in terms of the central nervous system being all the inner neurons. So it's going to be receiving information from sensory neurons, whether that's coming in from the special senses, the eyes, whether that's coming in from proprioceptors, and we'll talk about that especially in our sensory lecture. These are things that are telling us our position in space, telling us what the joint angle is, the amount of muscle tension in this particular area, what position I'm in, what muscle tension is found in certain muscles. All that information is giving us information about our body, as well as external information of what's happening on the outside environment. We've got all these receptors sending information in. That's the afferent pathway into the control center. And that integrative center, the integrative center with all those functions, whether it's thinking, decision-making, memory, perception of sensory information, processing of sensory information, is all taking place in the integrative center. And that's exactly what we're looking at in the in the central nervous system, that it consists of all these inner neurons that are going to be receiving information and processing it. And then, as the central nervous system, the inner neurons that are located in the brain and the spinal cord, they're going to now send information back out the efferent pathway. And the efferent pathway in that case are going to be those muscles. They're going to be the neurons and the glands that are going to be taking part. So we're going to be illustrating the pathways that are taking place in the central nervous system, just looking at those inner neurons. So as you can see in the bottom slide on the embryonic development, I'm just going to talk a little bit about this because the idea really isn't so much about what's taking place at the point at which an embryo develops is the rise of three different germ layers. These are different layers of the cell that we're looking at. There's an ectoderm, there's a mesoderm, and there's an endoderm. You don't really need to know that, so don't worry about that portion. But what you're looking at is when we look at a whole cluster of cells, they start to divide, and they're rapidly dividing. And as you look at the sphere that develops, look at a whole cluster of cells that just look like this. They're just rapidly divided, and it's a whole sphere, ball of dividing cells, that's eventually going to give rise to the developing fetus. And at that point in embryonic development, there's different phases or different layers that will start to form as differentiation starts to take place. It's going to start to create these different layers. So here's our sphere that we're just talking about. Now it's going to start to differentiate and create different layers where you have an ectoderm, an endoderm on the inside, and a mesoderm right in the middle. And what's happening is we're starting to give orientation to the fetus, where a certain part is going to be the head end, and the other one's going to become the tail end. And that's going to create one part, it's going to create the brain, and the other part's going to create the very bottom portion of the spinal cord. So what's happening at this point in time, and here you're seeing from a transverse view, just a cut right through the center, you'll see the folding of the ectoderm. And the ectoderm starts to fold where we start to see this portion right here. We create these neural folds, and then we start to see the arrangement of the neural tube. So what we're doing in this particular layer is we're creating a layer that becomes a long tube. So now if we take a look at that tube and take a look from the top portion of it, here's that view of that tube, that blue layer that starts to form. You see the formation of that plate, and then it wraps around and it's supposed to fold in itself to sort of create like a little tube. Okay? So as it starts to fold, you'll start to see areas of which this area starts to become the spinal cord, and this part becomes the brain. So if you take a look at the development of the fetus, you should see that these should fold all the way from the very bottom to the very end. Have you ever seen anybody with spinal bifida? Spinal bifida, you'll notice that they have very underdeveloped legs. In fact, their legs don't even really function. And that's due to the incomplete closure of the tail end. That plate doesn't close all the way, and that becomes underdeveloped. Essentially, if there's no central nervous system, meaning the spine isn't fully closed, then the legs don't work. Now, if you get incomplete closure of the head, well, then the brain doesn't develop, and then the fetus is ejected. And so forth. So looking at that, if you were to take a look at that tube, we've got this tube that's laid out for us, and we're looking at a bird's eye view of it, we're going to start to see that certain areas, particularly the head end, is going to start to develop a lot faster than the rest of the tube. And we can see this as the embryo starts to develop. Here we see about 3 to 4 weeks. We start to see about 7 weeks, 11 weeks, and eventually an adult brain. And what we're looking at is a regional specialization. We'll start to see an area that becomes the spinal cord, which is the tail end, and we start to see what's called the telencephalon, the front area, which is the head end. And as you notice, when you start to take a look at that embryo, as it starts to move through, you'll see that one area, specifically, you'll see in the book, it's called the forebrain, or the telencephalon, will start to develop much faster than the rest of that tube. The rest, earlier, you'll see the other tail end becoming the spinal cord, and this creates that profound development. So looking at from the side, from a lateral view, you can see that the telencephalon grows much further, and I'll show you that on the next slide. So here we're looking at that tube, right? We see that tube, that neural tube that formed, and what we're going to be looking at as that embryo starts to develop, you'll see that the telencephalon is the one that is going to give rise to become the brain that we would see, the adult brain. So this area is going to grow faster and faster, and that's something particularly interesting about embryos. If you look at embryos as a whole, and this is where they take a look at specifically embryonic, or at least developmental physiology, you'll start to see that embryos of many species look the same, especially early on. But what gives rise to primates, especially, is that the increasing telencephalon. This starts to grow faster and faster compared to the rest of the areas. And so areas of the spinal cord, the hindbrain, which is going to be the cerebellum and the thalamus, the midbrain, which is going to include the hypothalamus and areas here. These are going to be more of the primitive rat brain. And they're going to be more responsible for autonomic functions. Things like the heartbeat, respiratory rate, and so forth. The other areas, the telencephalon, that's going to give rise to the actual cerebrum, the bigger part of the brain, are going to be the higher mental functions that'll take place. Our capability of analytical capabilities, our conscious thought, our other higher level functioning. And so what we're going to be looking at next is just the telencephalon, how it grows. You'll be able to see on the next slide over, that same tube, not only are you going to see the regional specialization where you see the telencephalon growing faster, you can see that there's also lateral growth. Here you can see the left side and the right side. So this is going to give rise to the different areas of the brain, the left hemisphere and the right hemisphere. On the inside, you'll see that this is the tissue. Here's the space on the inside that's formed as a result of that tube forming. And that space is going to start to fill up with cerebral spinal fluid. We'll talk about that in the next later. So while we see growth of the telencephalon, we see the lateral sphere starting to grow. Here we can see the rise of what we're going to be looking at. So what you saw earlier, the blue part of the tube being the hindbrain, you're going to see is the cerebellum and the thalamus. We're going to see the yellow portion, which we saw, which is the midbrain, which typically is the brainstem. And then the biggest red portion is going to be the telencephalon, and that is going to be the cerebrum that we're going to be looking at. So now that you have an idea of how the telencephalon or the cerebrum is going to start to grow, the bigger areas of the brain, let's take a look at some of the other brain protection mechanisms for protection. And here you're going to see the meninges. And the purpose of the meninges is to serve as an outer protective layer for the brain. Of which, if you were to take a look at the meninges, you'll see that there are 3 specific layers. If you see the term meninges, you've probably heard the term meningitis. I know there was a question asking about meningitis. Meningitis is just an inflammation of these protective layers. Okay, it's a viral infection that comes in and can eventually make its way in. And there's a lot of concern for those viruses to eventually make their way into the neurons, so they can eventually start to destroy them. But typically, they only penetrate up to the outer layers. If you take a look at the layers themselves, you'll see that we have three main layers: the dura mater, arachnoid mater, and pia mater, and of which the term that you will see that's common amongst all these is mater, and that translates in Latin to mother. So the outer layer, the dura mater, bless you, dura mater, the durable mother, is the hardest layer of the outside. If you ever look at any dissection videos, and if you take a look at the brain, there was an outer layer that was the hardest portion. And you'll see that we have, with the dura mater, it's actually broken down into two separate layers, of which we have the endosteal layer, which is connected to the skull. You have the dural sinus, which is just a space in between the two layers, and then you're going to get the innermost layer, and this is going to be the meningeal layer. This is the layer that's actually almost close to the brain. If you take a look at the arachnoid mater, the next layer down, arachnoid mater, spider mother, you'll notice that the arachnoid mater actually has these trabeculae, which are these structures, and that allows things such as cerebral spinal fluid to flow through, which we'll tie in just a bit. And then the pia mater. Pia mater is actually pia standing for soft. Soft matter. This layer is going to have direct connection, or directly touch the actual brain tissue itself. So it's going to be the layer right over. And what that's going to do is going to have all these capillaries that are going to come in and provide nourishment for the neurons that are right up on that brain. And so what you're going to be able to see of all these, again, if these were the outer layers, you'll note that on the inside of the brain, we're going to also have that space, that same space that arose from the inside layer, from that neural tube. And that's going to fill up with cerebral spinal fluid. So taking a look at the four ventricles, those spaces that we talked about, the arrangement of those spaces, you're going to see we have four ventricles. Again, something that you already learned in anatomy. We have the lateral cavities over here, the lateral ventricles. We're going to have the third ventricle on the inside, which is going to be right by the thalamus. And we're going to have the fourth ventricle, which is going to be the ventricle out over here. And that area is going to be right by the brainstem and right in front of the cerebellum. So these areas are going to allow fluid to flow through. And where's that cerebral spinal fluid created? We know we're going to have what's called the choroid plexus, which is a whole bunch of tissue that consists of ependymal cells. And these are the cells, if you remember, as we mentioned. On the apical side, we're going to have cilia. And this is a single layer of either cuboidal or columnar epithelial cells that are going to be secreting stuff out into the lumen. So they are exocrine epithelial cells in that they're going to secrete out to the outer layer. And the cilia on the apical side are going to help brush that cerebral spinal fluid out. And you can see where that cerebral spinal fluid, if you take a look at the characteristics of the spinal fluid, it pretty much is very similar in composition to blood plasma. In that here we have a capillary that comes in, or specifically a blood vessel that's carrying blood. You're going to have our ependymal cells, which are going to have tight junctions in between. And they're holding very tightly so that things that that leave the blood have to flow through those epithelial cells. So they help govern and protect what's happening on the outside. And then as that blood flows through, now you're going to have what's called that blood-brain barrier. You have fluid coming back out, and that fluid is going to serve many functions, of which we're going to see mechanical protection. The brain is literally just bathing in cerebral spinal fluid. Okay? So you have this fluid on the outside that it helps absorb shock. Not only that, you'll also see that the brain, specifically the cerebrospinal fluid that's flowing through, is our direct pathway to tell us what's actually in the blood itself. So if I've got a bunch of acids in this area that's flowing through, acids such as lactic acid as a result of anaerobic metabolism, as a result of glycolysis. So lactic acid starting to form in that area. Now here you see that the blood, as the blood is flowing through those capillaries, we'll note a certain area called the medulla oblongata, which is going to have chemoreceptors. And we'll talk more about this in our sensory lecture after lecture 1. These chemoreceptors are detecting the levels of chemicals, hence why they're chemoreceptors. They tell us how many acids are in the cerebrospinal fluid, and that's going to be a good indication of how the rest of the body is doing. So it's literally just a dipstick just telling us, should we start to get rid of some of those acids? Should I start to increase some of the organ systems that are going to help us remove those acids in place? So these chemoreceptors are literally just detecting the pH of the cerebrospinal fluid, and that's going to let us know if the state of our body at that point. So now that cerebrospinal fluid flows, we know that as that fluid is flowing through, it's again similar in composition to plasma. We know that the choroid plexuses, as you can see here, these are the ventricles, the spaces that are filled out. You'll note that the choroid plexuses are found in all four of the ventricles. So these areas are going to be producing the cerebrospinal fluid that, as we saw earlier. And that flow, where that cerebrospinal fluid flows, you'll note that it follows that pathway from the lateral ventricles into the intraventricular foramen, into the third ventricle. Then it will flow down the cerebral aqueduct right in between the third and the fourth ventricle. So once it makes its way into the fourth ventricle, then it's going to make its way out in the subarachnoid space, down to the spinal cords, all the way down, and then make its way back up to the subarachnoid space. I know it looks like it's being mentioned twice, but I've got a video that will show you the flow of that. And the final destination is the arachnoid granulations, where it can make its way back into the venous system to be absorbed back into the bloodstream. So let's play this video real quick. That will show you what's taking place. So here we're just zooming in. We're going to start to take a look at the ventricles on the insides. Here, that space. And here we're looking at from the back view, you can see the lateral ventricles, third ventricle, and the fourth ventricle. Here you can see the subarachnoid spaces. These were the spaces that are filled with the cerebrospinal fluid. And we're going to point out how we get fluid to flow out to those areas. So we'll have to start with the ventricles, which we're going to see the choroid plexus, which are present in all four, all lit up in green. And they're going to be secreting fluid that's going to start building up in the ventricles. And that's going to create this flow that's going to move out from the lateral ventricles. I'll show you the pathway down to the third ventricle, down the cerebral aqueduct into the fourth ventricle, and then it's going to start to fill out that space in the subarachnoid space. Okay, out to the spinal cord, back to the subarachnoid space, out to the arachnoid granulations, where it's going to flow back out, back to the heart. So this is how we create that circulation. Now, this next slide shows you what happens if there's a blockage of that. And where that blockage would typically take place, typically takes place around in the interventricular foramen or the cerebral aqueduct. Any way you look at it, any blockage of that area is going to start to create a buildup of fluid. And that buildup of fluid, especially in babies, is called hydrocephalus. And this is where we see the blockage of cerebral spinal fluid, and it creates this enlargement of the brain itself. Nothing wrong with the brain tissue per se in the very beginning, but just an enlargement of the ventricles that causes that backup and expands the brain. And if you take a look at the baby's skull, we know that it's not fully fused. So because of that, you'll start to see an expansion, and this starts to pose an issue for the baby, especially when that brain when those ventricles fill up and start to compress the brain against the skull. Okay? So what they need to do in this case is they need to create a shunt. And what a shunt is basically a drainage pathway where they can lead it right into the lateral ventricles. And you'll note that that fluid now drains back out and makes its way back into the nose, back into the stomach. Why don't they just eliminate that cerebral spinal fluid? Well, baby's constantly making cerebral spinal fluid, and they need to get it back into the system. So now they have a drainage way so that it can make its way back and replenish its own fluids. So So that's our case of hydrocephalus. Let's sort of take a look at the brainstem. So now we're going to work our way from the hindbrain to the midbrain over to the forebrain. So let's start with talking about our primitive primitive areas first. And I call it primitive rat brain because the brainstem is really the area that's governing a lot of our primitive functions. So first looking at the medulla oblongata. Let's first start off with the cardiovascular center and respiratory center. We'll go into these when we talk about our cardiovascular lecture as well as a respiratory lecture. But the medulla oblongata is going to contain neurons that are going to tell us the state at which our heart is beating and which our lungs are working, giving us an idea that sensory information that's coming from that area to govern those areas. You'll also note that we also have what's called the decussation of the pyramids, contralateral tract crossing. You've probably heard at some point that your left hand is controlled by the right side of your brain. Okay? And then conversely, the right side of your hand or right side of your body is governed by the left side. So information crosses, any sensory information that happens in my hand crosses through to my left side, and all that efferent information starts from my left side and crosses through the right side. So the question is, where does it cross? It crosses right at the medulla oblongata. So that is what we call the decussation of the pyramids, where you see the term contralateral. Contra meaning opposite, lateral meaning from one side to another, or on one side. So contralateral crossing. So about 90% of the connections that you're going to be able to see will cross through the medulla oblongata. Any sensory information that's coming in as well. Let's say you pricked your hand. That information is going to flow from one side, cross through, and cross through the other side. Now looking at the midbrain, is also going to contain areas such as the substantia nigra. And this particular area is an area of the brain, and substantia nigra means black substance. And that particular area also contains dark nucleated tissue, meaning there's tissue that contains a lot of nuclei. So here we would typically see a lot of cell bodies that result in the production of dopamine. And these areas govern subconscious motor activities. So what am I thinking about subconscious motor activities? Motor meaning motion, subconscious. Can you think of any activities that are subconscious that you do? Breathing, okay. How about actually like motion?
I move my feet. Like you move your foot all the time.
When you move your foot?
Yeah. Like, I don't know.
Like restless leg? How about something a little bit simpler? How about on your way over here? Were you thinking about how to walk? Do you have to okay, now I gotta move my knee, I gotta lift up my leg? No, you didn't think about that. Those are subconscious motor movements. These are things that are sort of pre-programmed, things that you learned at some point. So they've become subconscious. So these things, and even thinking about certain, and let's tie in restless leg syndrome. Restless leg syndrome, when you look at these things, certain things such as Parkinson's disease create unwanted movements. Okay? And typically these areas, when you look at Parkinson's, you start to see that there's a lack of dopamine. So they'll start to give these patients L-DOPA, so forms of synthetic dopamine to help regulate the amount of that type of neurotransmitter. This area is also actually known as the reward center of the brain. So these are areas that are typically centered around addiction studies. They want to look at what's happening to these types of neurons that will result in somebody being more addictive or having that addictive personality. The next thing we'll look at is the cerebellum. And that cerebellum is this butterfly shape right at the very back. And in this area, by looking at this particular area, it is going to contain most of the neurons. So if you were looking at the cerebellum, it would look gray. So one thing we want to mention, gray matter. Why was it, well, let's take a look at white matter. What would make brain tissue white? I'll give you a clue. It has something to do with some fatty substance. Myelin sheathing. Myelin is white. And so if you were to take a look at where the axons are located, in the central nervous system, you'll notice, and this was a cut off of the brain right here, you'll notice that the outer layer of the brain will be gray, while the inside will be white. So white matter is going to contain the myelinated axons of the neurons, while the gray matter, and it's gray because it contains the DNA, contains the nuclei, it's typically darker, it actually contains the cell bodies, it's going to be on the outside. So in the CNS, gray matter on the outside, white matter on the inside. Now the neurons that are located, you have all these neurons and all cell bodies that are located in the cerebellum. Looking at from an afferent point of view, we have sensory information that's coming in from all portions of the body. Here you'll see the term proprioceptors. This is telling us about the position of the body, position of the joints, muscle tension. It's sending information over to the cerebellum. And then, on an efferent pathway, it's going to help. If it's sensory information saying, this is what I'm actually doing, cerebellum's going to say, this is what you should be doing. Okay? So what it's really doing is it's help maintaining quality control. And it helps balance what is the intended movement versus what's actually happening. Okay? So what's actually happening is sensory information being sent to the cerebellum. Cerebellum's going to correct that information and send it back out. So hence the subconscious skeletal muscle movement. So what you would typically see in the cerebellum is it helps maintain fine movements. So one thing I brought up with alcohol, just to kind of tie in what happened in last week's lecture. We talked about alcohol coming in and inhibiting some of the areas. And the areas that would inhibit were the cerebellum. And so if the cerebellum is responsible for muscle movement, and alcohol comes in and starts to interfere with those areas, it's not uncommon that you would see somebody that has high alcohol levels to not be able to really move as they intend. So that's why you would typically see somebody stumble or fall or just sway back and forth. And what's happening in the cerebellum with alcohol is that now we're inhibiting the cerebellum, and that's going to start to result in unintended movements, which the cerebellum is supposed to correct. So this is typically where you would see, and to give you a good example of what takes place in this quality control is when I scan the room. So if I'm looking from one side to the next, my intended movement is to look from left to right. And what actually is supposed to happen with the brain's program is sort of this movement like this. It's not actually smooth. What the cerebellum does is it's going to correct that sort of jagged movement as I'm scanning from left to right to make my eyes move in a smooth pattern. And if you ever, and let's hope you don't, but if you ever pulled over and a cop shines a light in your eye and they want to say, I want you to just follow my eyes real quick. What they're looking for is you're supposed to have smooth movement. Your cerebellum is supposed to correct that movement from left to right. But alcohol comes in and starts to inhibit certain areas of the cerebellum. So what's going to happen is with inhibition of the cerebellum, I don't have that quality control. So now my eye starts doing this as it moves across. And when the cop starts to see that jagged movement, they know that alcohol is involved, and they give them a reason to suspect that cerebellar functioning doesn't seem to be working as optimally as it should. Is there any sort of inebriation? Should we test for this? So that's one of the things that they measure on this, and that saccadic movement is this right here. Is what we call horizontal nystagmus. The next thing on here that you're also looking at is a condition called ataxia. And again, another case where this man has decreased cerebellar function, meaning the cerebellum cannot govern what he's actually doing. So as he's walking around, he's getting that feedback. The feedback's going back to the cerebellum, but the cerebellum cannot correct for those actions. So, let me give you another example. Has anybody ever stumbled on anything? Like if you're walking along and you stumble on a rock or something like that, what do you typically do? It's even subconscious. You don't even really realize it. You actually correct yourself before you're like, oh, wow, I almost fell. What actually helped you correct that movement? It's a cerebellum. You knew what your intended movement was to walk, but something came up and interrupted that, sent that feedback to your cerebellum, and then cerebellum says, nope, this is how you correct it. Take place back over there, correct your walk, and you say, oh, great, now I know what I'm supposed to be doing. In this case back with ataxia, this man doesn't have cerebellar function, so what he has trouble doing is actually correcting what he's trying to do, is to walk. So his neurons, his sensory neurons are saying one thing, cerebellum's saying another thing, and so he has to consciously work on positioning his body. He has to be aware of what's taking place in order to be able to walk. Thalamus and epithalamus. Main thing about the thalamus that I want to get across is the relay center. That's one thing I want to take place. The rest you guys can read on your own. But the relay center is that it relays information, any sensory information that flows in, whether it's from the general senses, touch, anywhere on the skin, pain, or anything from the special senses, from the eyes, ears, taste, or olfactory senses. Actually, scratch olfactory. All that information flows through the thalamus so that it can get routed over to areas of the brain that are relevant. And see these areas, lateral, geniculate, medial, intralaminar? Go ahead and cross that out. We're not really that concerned about that. But this just goes to show that areas of the thalamus relay visual information, areas relay auditory, and other areas promote sensory information flowing in. So I'm not really concerned that you guys know which lateral versus medial. I want you to just get across that sensory information flows in. But the one thing that you do know is that olfactory does not flow through the thalamus. And again, something we'll talk about in our sensory lecture. That's an exception to the rule. In fact, olfactory is actually one of our more finer senses. It's actually going to flow in a different way. So again, stay tuned for more of that when we get to our sensory lecture. The next thing I want to take a look at is the hypothalamus. The hypothalamus is going to be the very beginning of our endocrine lecture. This is where we're going to be able to start from the very beginning. And the hypothalamus is going to contain neurons that are going to be receiving information. It's going to also receive information and can send efferent information that will regulate hunger, thirst, and body temperature. It's where it all starts. It's the center for homeostasis. This is the control center. So it's going to be receiving information. It's going to then tell us how we're going to send that information back out. And the information that's being sent out is actually going to be in the form of hormones. Hence why hormones being our endocrine lecture. And so these are going to have specific part of the hypothalamus, or actually all the hypothalamus is going to contain neurons that are going to create hormones that are going to make their way to the next area. And the next area is going to be the pituitary gland. So here the pituitary gland is going to be separated by, we're going to have a posterior pituitary and we're going to have an anterior pituitary. And again, we'll get more in depth about this in our endocrine lecture, but here we're going to be seeing the secretion of those hormones. While some of the hormones are being created in the hypothalamus, you're going to get the release of hormones that are going to take place in the anterior pituitary as well as the posterior pituitary. So starting from here, back out here, being out released into those areas. And we'll get into more detail about what's taking place. So in our endocrine lectures, we'll be talking about hypothalamus to the next target, which would be the pituitary gland. And if you remember from our first lecture, when we were talking about the secretion of testosterone, the next target would be the testes in this case. And pituitary, whether it's the testes, whether it's the mammary glands, whether it's pancreas, multiple targets for the pituitary. So the pituitary is going to send out hormones that will target other endocrine cells to start producing more hormones. The next thing we're going to look at is the reticular formation. And the reticular formation isn't necessarily a particular area of the structure of the brain, but what it is just a whole network of nuclei and nerves that are all tied into sensory information. So all that information that flows in from the thalamus is going to get directed out to their respective areas. So there's going to be visual information. Visual information will get sent back to the back area of the brain. If you're going to have information about motion, that's going to get sent over to the frontal lobe. If you're going to get sensory information coming in, that's going to come in more to the parietal lobe. So this is an entire network that's going to take in sensory information and route that over to where it needs to go. So the reticular formation is important because it does govern two things. And the first thing I'm going to point out here is habituation of constant sensory stimulation. And what do I mean by that? This is our conscious way of filtering out information that is non-relevant. For instance, this whole time, you've been sensing that air conditioner. You subconsciously have been hearing it this whole time. But it's not really important information to you, because hopefully you're listening to me talking. Okay? So you've tuned out, and that is what habituation is to turn off constant information that's being sent in. Your ears are always working, and they're always sending sensory information. But the reticular formation is making its way over to the inner neurons that will say, this information does not play any importance to me right now. I'm going to tune that out. So while the sensory information is coming in, the perception of it is not. It's not until I actually probably pointed out, you're like, oh yeah, it is on. I've actually come in and turned off the reticular formation for that particular sense, or that particular auditory information that's coming in. The next thing you'll see is nonspecific arousal of the cerebral cortex to incoming sensory information. So let me give you a scenario where you're waking up, and however you wake up, it's sad to think that most of us probably wake to an alarm, right? I don't know how many of you actually just wake up naturally and just be like, oh, it's a great time to start the day. But let's think of like Saturday morning. I don't know if you don't work on Saturdays. You want to wake up naturally. And typically, what happens when you're waking up naturally, you might start to become slightly aware of certain things. Like you start to hear maybe a chirp that happened outside. Or maybe you noticed, oh, maybe the temperature is starting to get warm on my bed. I need to get up. Or maybe, oh, my bladder's kind of full. I need to get up. All that is sensory information that your body is perceiving. And it's routing that information up the brainstem, into the thalamus, and it's routing it to the certain areas. And some of that information is crucial. It's going to start to wake that part of you up. So it's actually involved in the whole sleep-wake cycle, where you actually start to wake yourself back up, and it's sending sensory information so that it can now alert you of what's actually going on. What's also interesting too is how many people here drink coffee? Okay. How do you guys know about the caffeine now? No? Okay. Well, let me tell you about what happens about the sensory information that comes in. Sensory information that comes in typically going to involve adenosine as a neurotransmitter. And adenosine comes in and starts binding to the receptors and saying, I'm getting sensory information. And as you go through the course of the day, what's happening is adenosine is binding to those receptors, and the receptors over time stop to work as efficiently because they kept receiving that neurotransmitter. It's a form of habituation, but it's what's happening with the receptors themselves. And when you go back to sleep, the adenosine that binds to those receptors, they start to not attach anymore. Why? Because the reticular formation isn't working as much anymore. It's actually shutting back off, and those receptors are replenishing themselves, and they're waiting so that they start receiving more sensory information, and once those adenosine receptors receive information, then you start to wake back up. What's interesting about caffeine is caffeine binds to those same adenosine receptors because the active site is very similar. Or the molecule that binds, caffeine that binds to the receptor is very similar to adenosine. So what happens with caffeine? Now think about when you first drink caffeine in the morning, right? When you first drink caffeine in the morning, those adenosine receptors are kind of fresh. There's not much adenosine that starts binding because the pathways don't happen as much in the morning. So those receptors are less likely to be bound to adenosine, and they'll take in caffeine. Caffeine is the greatest when you first wake up, right? You boost up, and it starts to activate the reticular formation, and now you start receiving, and things start to ramp up. Now, over the course of the day, and you probably did this before you started class today, who here drank a cup of coffee before you started? How effective is that cup of coffee at the end of the day versus the very beginning? Not as effective. Why? Because what's happening is that all that adenosine that's binding to those reticular adenosine receptors, we've got a lot of adenosine already there that's bound to those receptors. Caffeine doesn't really have much of a place to bind. Therefore, it's not effective. So back to this whole thing where I was talking about a caffeine nap. If you want caffeine to be really effective, you just got to take a nap. Give your reticular formation a chance to rest. Let those adenosines unbind to those receptors. So what you actually end up doing, and look it up if you don't believe me, caffeine nap. It literally tells you, drink your caffeine, just pound your coffee, and of course don't pound it hot, but drink it fairly quickly and go take a nap for about 20 minutes. Because it takes about a good 20 minutes for the caffeine to make its way into the small intestine, to make its way back into the brain, and make it to the cerebrospinal fluid, where it starts to make its way into the brain tissue. Because it actually crosses the blood-brain barrier. Once it starts to make its way into the brain is when you start to wake up. Because then caffeine is starting to bind to the reticular formation, and when you wake up, you'll feel super refreshed. If you don't believe me, try it one day. Take a caffeine nap. And even think about this, it also really works. I used to do this before, was I wake up in the morning, I'd have coffee that was already made, I would make like cold brew, pound it, and I'd go back, take another 20-minute nap. And when I woke up, my heart was pounding, and I was awake, I'm like, wow, I'm ready to go. And I was like in and out of it, I was real quick, as opposed to that like, just waiting you drink that coffee and you wait, you're like waiting like 5 minutes, now I'm finally wake up. So this is your way of getting that boost. So if you ever need to, especially if you're cramming right before next Tuesday, try a caffeine nap. Try it out. What's that?
Can you tell our bosses that work now?
Keep that secret for yourself.
You go take a nap. Hold on, you go take a nap.
Just take a quick 20-minute nap and caffeine. Now the cerebrum is looking at the outside. So this is where we get the telencephalon. And again, being humans, this is where we're going to see the largest portion of the brain itself. You look at all the brain tissue, most of that is going to be the cerebrum. That's the outside of the brain itself. And what connects the left hemisphere from the right hemisphere is this band of white tissue called the corpus callosum. And that corpus callosum is going to have axons that are going to travel. Again, it's a white matter. It's going to allow axons that are travel from the left side to the right side. So there's a lot of connection between the left and right sides that are involved. And so what you're going to notice about the cerebrum, and again, the cerebrum being on the outside, notice how the outside is going to be gray matter, the inside is going to be white matter. Again, you'd expect the neurons to be located, or the cell bodies to be located on the outside gray matter, while you get the axons, which are myelinated, on the inside white matter. And you'll notice areas, what we call cerebral lateralization, where certain areas of the brain have more of a specific function as opposed to others. And so you'll note the left side of the brain happens to have more speech, writing, more analytics, as opposed to the right side, which has more visual spatial tasks involved. So things such as navigation, things like geometry, things like shapes and positions, as opposed to these areas, which are more related to language. So you may have heard the term somebody might be left brain versus somebody might be right brain. And that's related to these areas. Now, how do they know that these areas exist? Well, these come to some of the earliest scientific studies. What better way to find out what this area of the brain does? By poking at it. And they used to do them on people that were awake. They used to just literally cut the brain open, expose, cut they would, I don't think they would cut any of the meninges, but they would come in and start poking areas. And they would say, okay, well, this area, I want you to count from 1 to 10. 1, 2, 3, 4, 5, and they start poking at that area. And they start poking at that area, it starts affecting their speech patterns. And they'll just notice, yeah, I could think the numbers, but I just couldn't say it. Well, so that area must be speech. Start poking at this area, and all of a sudden, their ability to hear information starts to decrease. So this area must be involved. Start coming in and start messing with this area, and they start saying, oh, now the right side of my vision starts to get faded. And that area must be related to vision. So they start to notice that these areas have very specific functioning. In fact, it's not like this area is the only area involved. If you look at speech and language, you know that anything that's related to speech has to travel to that area for language comprehension. Typically, if you're listening to me talk, there's a lot involved. It's not just language comprehension, speech. It's being able to look at the language center and relate that to areas of the brain that have that area stored. So information is traveling back and forth, not only in between, not only on one side of the cerebrum, but also in between the hemispheres. And that's what the corpus callosum does, is it allows information to travel back and forth. And I'll talk about the pathways that lead to. Now, there is an issue with people with certain types of seizures. And these are fairly severe seizures, where one of the last measures that they can do is they can cut the corpus callosum. So what they find about seizures is that you get a lot of information and a lot of, I would say, nonsensical, extreme electrical activity that happens on both sides, causing seizures to take place. And what they can do to prevent some of the seizures is cut the area to prevent seizures from traveling from both sides. And that typically will mitigate the amount of seizures taking place. So these areas where they cut the corpus callosum can now prevent information from traveling from one side to the next. So the things where we mentioned earlier, left brain controlling the right side, that information can't travel to the other side. So it's not uncommon for people that have this what's called split brain, because we've cut the corpus callosum, to have almost two independent minds working. Yes?
Is that why with the stroke they have problems with speech and comprehending certain?
Yes, certainly, because areas of damage, well, depending on where that stroke takes place, can start to affect those areas.
So when they split it, are they still like a vegetable, or are they still?
You want to see what that looks like? Yeah, got a video for it.
Oh.
So this is an old video, but obviously you can tell by the equipment that they're using. But they're watching a guy who actually had his corpus callosum severed due to epileptic seizures. And what they end up doing, I'll just show you the video. We'll just kind of cut to, because it's a 10-minute video and I certainly don't want to play the entire thing.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire.
Don't mute.
Scientists might design storms that caused his seizures, but it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently. Do you feel any different when you think about something than you did any differently from the way you felt before the procedure?
It's just got a backup brain, that's all.
That's something everybody could use, right? I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we're saying that the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside-down duck. Okay. But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, it's really like two different people doing the same.
That's right.
Same task.
That's the idea.
Okay, Joe, I want you to keep your hands up.
In an experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Shit.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Store.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano. Good.
But when a word is flashed to his right brain,
Didn't see that.
Okay. So I'm going to ask you
but now watch what happens.
to draw that with your left hand.
Or visuospatial. He wasn't able to verbalize
I think
it.
I'm a little lost.
Why don't you try drawing another picture of it over here? That will help you.
Awful.
Oh, okay.
Sure.
It's almost as though somebody has given him a secret communication.
That's right.
Now he knows that it's a telephone. Like, up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name
You
it.
said church, didn't you?
After looking at the picture.
Yeah, but he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
The communication is not occurring inside the head, it's occurring out on the piece of paper.
Yeah.
Blob. I don't know.
You want to draw a little bit more?
So far, Joe has been seeing only one word.
That's
Things get even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere saw toad.
Yeah.
And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad, right?
And this time I was able to guess what was coming.
Will we put a little three-legged stool in there later, or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret.
That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly the point. I've been doing this for 35 years.
Yeah.
And
It gets me every time.
Yeah, it must.
This time, instead of naming the word, I want you to point to the word.
Again, Joe sees two words simultaneously. Bell goes to his non-speaking right brain. Music to his speaking left brain. When asked to point to a picture of what he saw, he chooses Bell. But when asked why,
Okay, why'd you pick that one?
Music.
Music?
and when asked to explain,
There was music, bell, and it was a few minutes ago, last time I heard any music was coming from.
Yeah. So that is a long video, but it goes into some of the things that he ends up going into. So what you saw was a split brain where one regional functional area of the brain cannot communicate to the others. So you can see at least the hindrance behind that, why we have the corpus callosum in the first place. So certain memories can't be associated. So any visual things that he can't see might not be able to even really vocalize what it is.
And he has split personality.
What's that? I don't know. So looking at the cerebrum, you start to see different areas of the brain. I'm just going to kind of whip through these. The cerebrum, if you take a look, is actually divided. And one couple things I want to point out is why this wrinkling that's taking place that you would typically see in a brain. Well, this is why the telencephalon grows so fast. In the skull itself, it doesn't have enough space, so it starts to create increased surface area. So we start to see areas that are divided, where we're going to be able to see areas that have these fissures, which are going to be called sulcus or sulci, and here I'll have all of them defined for you. And areas that are prominent folds are going to be called gyrocytes or gyri for plural. So we're going to start to take a look at areas of the brain that are divided from the frontal lobe, parietal lobe, and they're all divided by main anatomical features of the brain. So the central sulcus is going to start to divide some of these areas. You're going to see the occipital lobe towards the back, temporal towards the medial sides of the brain, and the frontal lobe in the very front. So with that, we'll be able to take a look at some of the functions. The frontal lobe is going to contain areas of the frontal lobe, when we look at it's mostly involved in, you'll see areas for emotion, but you'll also see motion and motor control. So these areas you would look at that we define right by the central sulcus. The most posterior portion of the frontal lobe is going to be called, where am I? The precentral gyrus. So these are going to contain the motor neurons, meaning that these are going to contain areas that are going to be tied into areas for motion. And this area, the parietal lobe, the more anterior portion, is going to contain, and this is going to be called the somatosensory area. This is somatosensory. And this is primary motor. Somatosensory is going to contain neurons that's going to be receiving information about the body. So sensory information about the soma being body, sensory receiving sensory information. So this area will start to receive information while this area will govern movement of those areas. So one of the things that we'll talk about later on is the areas of the brain, or areas that's going to resemble the brain, and you'll be able to see that on a later slide. Areas that involve receiving the most sensory information. It's going to represent the largest areas of that gyrus. And we'll talk about that in a little bit. The next slide I have over, I'll leave this for the very end. You don't have to see it now. But I'll leave that for the end that we can see later on. I have a video all about the lobotomy. And one of the things that they do for a lobotomy is to sever the areas on the frontal lobe to limit the amount of activity that's taking place in the emotional centers. And so they typically would do this. I'll talk about that in just a little bit. This is what's called the somatosensory motor homunculus. So here we're looking at the primary, this would be the parietal lobe. And this area would be the frontal lobe. And that's all divided by that central sulcus, right in the very center. So here it would be the motor, this would be somatosensory. And notice about the areas that resemble, or the areas that represent the areas that are most sensitive, such as the lips, the hands, the tongue, and the face. These areas contain the most number of neurons that receive information from that area. So they are going to comprise the biggest slice of that gyrus itself. Areas that receive the least amount of sensory information. Okay, so things like the hip, things like the teeth, gums, and maybe not teeth, gums, and jaw. Looking at the elbow and the arm. Those areas don't receive as much sensory information, so they're going to have the smallest slices of the motor homunculus. So what you see that's drawn on the side is the areas that represent those areas of the body from receiving sensory information. Now similarly, on the primary motor, again, the more posterior region of the frontal lobe is going to contain the same neurons that are going to be responsible for motion. So these are going to contain the neurons, the cell bodies for what's called the upper motor neurons. These are going to be neurons that are going to travel down. They're going to travel down to their respective areas. So areas that require the most muscle control are going to contain, again, the largest sections of this brain slice, okay, of this gyrus. So areas like the hand, which requires a lot of motor control, lips, tongue, all that, all responsible for speech, facial muscles. So face and hand have the most motor control. So you're going to see more neurons of those areas. Areas such as the knee is going to represent a very small portion. Because either the knee can flex or it can extend. It's not really much. Yeah, sure, there might be bigger muscles, but less control required from the cerebrum itself. The other area is occipital lobe. That's going to be for vision. Insula, typically for memory. Insula would be more the inside of the brain itself. I'll leave you guys to these. Cerebral white matter, again, taking a look at the white matter itself. Cerebral white matter is just looking at the axons. So this is not the gray matter, which is the outside, the white matter is going to be the axon bundles themselves. And if you take a look at the axon tracks, these are things that are traveling from one side of the brain to the other, or traveling within the same hemisphere. These are inner neurons that are starting from one neuron and targeting another neuron. So these neurons, these tracks of nerves or axons can all be labeled as three types of tracks: association tracks, commissural tracks, and projection tracks. Association tracks are just things that travel on the same hemisphere but to different gyri. So same side, just traveling all throughout from one side to another. Now commissural tracks travel from one gyri and to the different hemisphere. So naturally, these are going to have to cross through the corpus callosum. So when that guy saw earlier, when he saw a picture of it, he saw the word toad, it needed to travel through the other hemisphere so that he can actually visually see it. So the last one would be the projection tracks. That's going to start from the cerebrum and travel down the brainstem out to the spinal cord. So these are going to project further back out. So each of these you would typically see, and here you can see some of the track bundles of where they go. Commissural traveling through the opposite side, through the corpus callosum. So here the corpus callosum, you would typically see projections back out. Projection tracks that travel down the brain, and then association tracks that travel from the same side. The last one is the basal nuclei, and looking at these are going to be responsible for voluntary movements. The basal nuclei are just, again, clusters of neuron cell bodies that are located within the brain itself. And if you were to cut and look at this transverse slice, you'll see these prominent features right here. And that's exactly what the basal nuclei are called. It used to be called basal ganglia, but now the technical term is basal nuclei. And these are really involved in voluntary movement. So it's things like the, that you're in direct control of. We'll finish up with visualizing the brain. The last thing I wanted to leave with is, if you guys are particularly sensitive, and this is my disclaimer, if you guys are particularly sensitive to watching some of these procedures, this one isn't necessarily gory per se, because it's a really old video, but this is what they used to do for lobotomy. So if you feel uncomfortable watching this, it's not a very long video, I think it's like 3 minutes, but it's what they used to do for people with certain mental diseases. Is they used to, before they even knew any better, was to find a way to actually punch a hole into the brain, into the platform itself, and sever areas of the frontal lobe. So this one they call, going back to it, this was actually done by a, back in the 40s and 50s when they used to do lobotomies. And this is an example of transorbital lobotomy, where they were able to go in through the eye socket and sever certain areas. And so what they did as a form of psychiatric treatment was to cut these areas. But unfortunately, while it was able to maybe, quote unquote, cure their psychosis, it resulted in a lot of other adverse events, things such as a loss of personality, loss of motor control in some areas. And so in this time, they only really knew the shape of the brain, and they knew they just needed to cut those projection tracks and cut some of those axon bundles. What they would do is they knew where the positions were, so they took a needle and they would literally inject it in and just know how to spatially, where that area they needed to cut was. Let me just play the video and you guys can see how that's played out. So we're done with the lecture at this point, so if you want to pack up, if you feel like going, that's perfectly okay. If you're interested in wanting to watch the lobotomy, stick around. Again, not very long, but a little gruesome.
The hand with the fingers extended demonstrates in 3 dimensions the course of the fibers from the thalamus to the frontal lobe. The thalamus is represented by the wristwatch. The fibers to the motor area go upward like the thumb to the convexity. Those to the frontal pole run forward like the second and third fingers, while those to the base curve around the anterior horn of the ventricle.
What's interesting
like
was
the fourth and fifth fingers.
The Portuguese neurologist that studied this won the Nobel Prize back in 1947.
Sever the fibers to the frontal pole by the lateral and medial cut, and then to sever those to the base of the frontal lobe by elevating the handle of the instrument. The connection between the thalamus and the frontal lobe are clearly shown in this illustration from Plexig's monograph on myelinization of the infant brain. Projection pathways from the thalamus to the occipital and temporal lobes are well myelinated, as well as those to the frontal projection. Beyond the anterior horn of the lateral ventricle, the radiation
Yes,
bends
they did
rather
talk about
sharply
it.
toward the midline and a sort of elbow. It is the aim of transorbital lobotomy to cut across this tract of fibers close to the elbow. Turning now to the operation itself, very little preparation is necessary for transorbital lobotomy. This patient came to the hospital this morning after breakfast, and if all goes well, she will leave tomorrow afternoon. The operation can be done under any general anesthetic, but electroshock is preferred because of its familiarity to the scientist. Transorbital lobotomy is performed during the stage of post-convulsive coma. The electrodes are applied and the first shock is given.
So they're giving ECT, electroconvulsive therapy. Shock that area.
The convulsion lasts about 40 seconds. When the patient shows signs of returning consciousness, a second convulsion is administered. Usually 3 successive convulsions are necessary, but in old people a single one may be sufficient, while in a sturdy young person 4 or even 6 convulsions may be administered without danger.
And here they're just triggering their seizures to take place. They still do ECT, but they obviously have very refined the procedure where it's
Now that
not
the
causing
convulsion
major
has subsided,
convulsions.
the nurse holds a towel over the nose and mouth of the patient. The operator lifts the upper eyelid, inserts the leucotome into the conjunctival sac, and aims it parallel with the bony ridge of the nose. He drives the point through the orbital plate, and at a depth of 5 centimeters, swings the handle far laterally.
Or no?
He then returns the instrument to a slightly oblique position, still parallel with the bony ridge of the nose, and drives it 2 centimeters further.
It's why they promote the seizures in the first place, so that they're kind of unconscious.
How's that relation like when they accidentally wake up?
Steadying the patient's head,
Probably.
he then moves the handle of the instrument about 20 degrees medially and 30 degrees laterally. In this latter position, he strongly elevates the handle of the instrument, often fracturing the orbital plate. And then finally returns the instrument to the parasagittal plane. The operation on the left side is seen from the profile view, which best illustrates the deep frontal cut.
Do the other side. Yeah, they bring gloves.
Oh, she did the box.
So they're not severing the eye, they're just getting right above that space. And it's funny because they considered it sort of an outpatient procedure.
This outpatient? They're doing the clinics?
It's like out the next day, pretty much.
There's clinics.
Obviously very controversial. It became more controversial, and obviously they stopped doing these.
Do these people volunteer to do this, or?
I don't think they volunteered to do it. I think it's more of this sort of clinical course of treatment. And they just let them know that, we don't have any other options for you other than a lobotomy, and it's really just.
It will be noted that the instrument
risks,
upon
the
removal
benefits outweigh
appears
the
clear
risks.
and shiny, but in view of its contact with the skin of the lower eyelid, different instruments are used for operation on the 2 sides. In this second patient, after operation, there is no damage visible.
Well, thank you for your guys' attention. Thank you for sticking around. Our neurons all the way out to our action potentials. So you should now understand how the neurons are firing. What we're going to be doing today is to elaborate more on the central nervous system. As we mentioned, the central nervous system is filled with interneurons. And those interneurons are all part of the integrative system that is doing all the thinking, decision-making, memory, perception. It's receiving sensory information from our senses, and it's going to be sending motor outputs to either through the autonomic pathways or through the somatic pathways. So we're going to start learning towards the end of today We'll learn about the spine and the efferent and afferent pathways that travel through. But right now I just want to kind of talk more about really just the brain before we move into the spinal cord. So when we talked on Tuesday, we talked specifically just about the brain structure. So that isn't very conceptual. It's really just going through and talking about the different features, different areas of the brain, and particularly the functions that they typically exhibit. And one of the things the key things that we talked about, we looked at cerebral lateralization, in that areas of the brain have, whether you're looking at the left hemisphere or the right hemisphere, have regional specialization in terms of its function. So if you look at the left hemisphere, this is more related to analytics. You look at calculations, you look at speech and writing, as opposed to the right side, which is more visual spatial, meaning looking at spatial concepts, being able to map things in your head. So people that are really good at looking at maps, or not even really looking at maps, people that drive places, and they just generally know where to go because they can see it in their head, those are very right-brained people. Okay? Now, when you look at certain damages to certain areas, and I know somebody brought up mentioned earlier, that's areas of stroke. Depending on where the areas of the stroke can really affect, you can tell will affect that particular area. So any damages to the left side of the brain, more towards like the temporal lobe, frontal lobe area, may have speech problems. May have difficulty speaking afterwards due to the damage in that particular area. And then any areas to the right hemisphere may start having difficulty trying to find their way around. So may have a tendency to get lost because they've lost that ability to map things out. And so we looked at our split brain experiments, and I'm going to skip into the areas that we talked about. We looked at the central sulcus, which splits the frontal lobe and the parietal lobe, and we looked at two particular areas. The front area of the central sulcus, which is the primary motor cortex, is the area that's responsible for motor. Okay, so this is where you can control area, where we're going to see what we're going to know later on as upper motor neurons. These are the neurons that are going to be sent over to the spinal cord segments that will control that particular area of the brain. And I'll talk more about that. I just want to do a quick recap. Now, the parietal lobe, the most frontal part of the parietal lobe, is called the somatosensory cortex. So this whole band right here is the one that we're talking about. This area of the parietal lobe is responsible for somatosensory information. That's information that's sent in, sensory information sent in from that particular body part. So when we looked at the somatosensory motor homunculus, this was what I was talking about last time, was at the tail end of Tuesday's lecture, is that certain areas, when we look at the primary motor cortex, here you can see that split with the central sulcus, right at the front is the primary motor cortex, areas that require, specifically for motor, areas that require fine motor control. Lots of muscles in that area that require a lot of control, such as the face, the lips, the neck, well maybe not the neck, but mostly the face and the hands, represent more areas of that slice. Okay? As opposed to other areas where, yes, they may be bigger muscles, but it's pretty much the same gross movement. I mean, flexion of the knee, and that's really all it's doing. So those areas have a tendency to have a very small portion of the motor cortex. Same with the somatosensory, so areas that receive a lot more information, such as the lips, hands, and so forth, have a lot, represent a big area of that slice, because it receives a lot of information and sends that back. The areas that we talked about can send information back and forth from each other. So we'll talk about that when we look at our reflexes. So with that, I want to start to tie into what we're going to be looking at. We finished off with some of the other functions, the cerebral white matter. Remember, we talked about the gray matter being the neuron cell bodies on the outside, white matter being the myelinated axons on the inside of the brain. So now let's get into more some of the functions of the brain itself. And when looking at the brain, there's really no way aside from just going in and actually looking at it. We do have different clinical ways of visualizing the brain itself, the three of which you're probably already familiar with. And the first portion is called a CT scan. This is typically something that is done. It's a quick X-ray, and what's typically done with a CT scan is are X-rays that are sent into the soft tissue, and these X-rays are absorbed based on the density of the tissue, whether we're looking at bone, if we're looking at muscle, or we're looking at fat, or even air pockets. Areas that are typically denser will absorb more of the X-rays, while areas that are fairly lighter, like areas of soft tissue, may not absorb as much X-rays. So therefore, when you get these X-rays going in, you can see the different densities of tissues, and you can tell if there's any particular masses or anything like that. That's usually the first thing they do. It's a fairly quick, easy scan. The next one is quite a different type of scan called the positron emission tomography. And basically what this comes down to are the use of contrast, radioactive isotopes. If you remember from our first lecture, we talked about contrast that's used. And this was our tagged fluorodeoxyglucose, FDG, which is a common contrast that's given in PET scans. And that is a tagged glucose molecule that will eventually make its way over to areas that are fairly active. And what they're looking for in a PET scan is to look for areas that have high metabolism. Okay? So how does that work specifically? Well, these isotopes that make their way out, they emit these radiation that comes off that contrast, specifically off that FDG, and it interacts with the electrons. So any cells that have high electron activity are going to get lit up, and then that is actually seen in the imaging. So think about where you would see electrons. Where would you see electrons in active cells? Think about that for a second. This is you're going to have to go back to a prior lecture. Where did we see cells that were generating a lot of, I don't want to give it away, but highly metabolically active, meaning it's using a lot of glucose. And where do those electrons come from? Where do you see a lot of electron activity? I know you know the answer. You got to think about that.
Does that have to do with movement?
Well, think of electrons moving around a lot. I'll give you another clue. It's somewhere in the mitochondria.
Oh, ATP.
ATP, okay, you're getting there. But specifically, which area of the whole ATP cellular respiration bit?
In the
membrane.
potassium process?
Almost. It's in the name. Electron transport chain. So if you remember that was our first cellular respiration lecture. We had those enzymes. We had NADH and we had FADH2 that are donating those electrons, and they're moving from enzyme to enzyme in the electron transport chain. So these things are moving back and forth, and they're constantly generating ATP. So there's a lot of activity in the mitochondria. So as those electrons are moving around, contrast is sitting there hitting it with positrons. These are just basically gamma energy that are crashing with those electrons, and they're lighting up, and they can see areas of the brain that are active. So this is particularly useful if they're trying to do like a functional scan where they are looking at areas, or if they're looking at areas that are growing way too fast. Right? So things like cancer and so forth, or if they're looking at drugs, for instance, and they want to find if the drug is being effective in a certain area. And kind of taking that back I'll talk about that later. The next one is the MRI, so magnetic resonance imaging. So this is another scan where obviously no contrast is used in most cases, but what they would do is they use these huge magnets. Has anybody ever been in an MRI before? Yeah. Okay, so an MRI is just basically a small tube, and you've got these huge magnets that are moving around. And what they do, as these magnets move around, it creates the protons, the hydrogen ions. Obviously being used quite a bit in the mitochondria. You saw the chemiosmotic mechanism. Those protons move around quite a bit, and that creates areas, and that allows us to be able to visually see it. So you get these protons that are vibrating, and you can then therefore see the imaging that takes place. And that gives us a really good view, because we can see from slice to slice, and we can actually see each of the physical features based on that. Right. So the next thing we can do, so that gives you imaging of the brain itself. Now we can go in and actually measure the electrical activity that takes place in the brain. And this is what we call the electroencephalograph. So if you probably at some point have seen EKGs, so the electrocardiograph, these show electrical activity of the brain. And so this is kind of an exaggeration. Nobody really for an EEG, nobody wears this many probes onto their head. But what we're doing is we're measuring the action potentials taking place in the brain. Where is it taking place? It's taking place in the cell bodies and dendrites. We're looking at, well, mostly in the axons. The action potentials that are taking place, we can measure those. And what we're looking for in an EEG are different waveforms. And I'll show you that in a little bit. Anytime you get the movement of these ions through the neurons, it's going to produce a little bit of a current. So we're looking at changes in those currents. And the purpose of this is that typically what we want to do is just measure the waveforms, which we'll look at on the next slide. But what's interesting about these brain patterns is if you were ever plugged into an EEG machine and you look at the waveforms that are produced out of here, they're going to sum up all the movements, all the electrical activity in the brain, and you'd probably venture to think that it was rhythmic, but that's actually not true. Ever looking at an EEG, you would see that our waveforms are actually very abnormally regular, meaning it looks like, I'm just going to draw a bunch of scribbles. It just looks like this. You just get a bunch of random activity. And that's because there's a lot of spontaneous activity that's happening in the brain, and there's really no rhyme or reason behind it. There's just so much firing going on. And that's just you being conscious, being awake, and receiving information, constantly receiving sensory information. So there's no one particular area that's going to exhibit more electrical activity than another. So let's take a look on the next slide on particular EEG patterns, so electroencephalograph. And we can measure different types of waveforms, 4 that I'm providing for you right here, that characterize the amount of electrical activity that's happening in the brain. And the one that we're all experiencing right now are beta waves. This is our first wave. And before I start to get into the waves, let me just kind of go over some of the real basics of wave patterns. And so what you probably may have remembered are wave patterns that look like this. And we know that the wavelength is the distance from one part of the cycle to the next. And you can see that this is constantly repeating. We get repeating 1, 2, 3 signals right there. We have the amplitude, which is the height of the wave. And then what we can do in terms of measuring how many waveforms we get, we basically measure how many waveforms we get per second. Okay, so we get a lot of activity and you measure out, this is the length of a second. You get 1, 2, 3, 4, 5, 6 cycles per second. And that also is referred to as hertz. It's also the same thing you've heard about radio signals. How many times do you see a wave, each repeating wave every second? So naturally, when you get higher frequencies, you would start to see something more like this. Higher amplitudes, you can see higher waveforms. Smaller waveforms, smaller in amplitude, just like that to the right. So now that we have that down, let's take a look and see the patterns. So like I said, all of us are experiencing beta waves right now, which is basically us being awake, simply awake, and we're receiving information. You're seeing visual stimuli up on the PowerPoint, you're hearing my voice, you're thinking about things, so forth. Where do you see most of that energy coming from? Where do we see where it's mostly characterized? We see it in the frontal lobes and the precentral gyrus. Okay. If you were to take a look at where that activity happens, or how much activity, it's about 13 to 25 cycles per second. And for the purpose of the test, I'm never going to really ask you about the number of cycles per second. I just want you guys to get the general gist of it. So these waveforms are small amplitude, right? Very small amplitude and fast. So as we move down from beta to alpha to theta to delta, you'll see we're going to be going slower. So these are going to be slower cycles. So we're going from low amplitude, high frequency, down to low frequency, higher amplitude. So Beta waves here, you can see what the waveforms look like. Very small amplitude, high frequency. These are typically things that you see with concentration, arousal, being conscious, being awake. The next thing to look at are alpha waves. And alpha waves are typically elicited when you're just relaxed. Close your eyes a little bit, relax, try to think about really nothing. And that's going to start to evoke alpha waves, where you start to see the waveforms start to slow down. And as those waveforms slow down, you'll see the amplitude start to raise. Okay? And that's because you get a lot less competing waveforms over here. Once you start to get the waves more uniform, you'll see that they start to go a little bit higher. And where do we typically see those? Parietal and occipital lobes. If you were to where you would measure alpha waves is somebody right before sleep. You actually get it when during meditation, and you also get it from watching TV. So if you sit there and you kind of just zone out on the TV and you don't really think about anything at all, measure it, you'll see a lot of alpha waves happening. The next one are theta waves. And theta waves, if you were to see where those take place, really in the back of the head, in the temporal region. Theta waves, again, slower but higher amplitude. And with these theta waves, they are fairly significant, and you would see that this is typically something that happens during sleep. It actually happens during REM sleep, which we'll be talking about, rapid eye movement. A lot of theory behind what's happening with our theta waves in regards to memory, emotions, how we process information, and things that happen during sleep. And again, I'll go more into what happens in REM sleep at a later phase. And then the last one are delta waves. And here, delta waves, you can clearly see really high amplitude and very slow waves. Here you can see about one cycle, about 1 to 2 cycles per minute. And you'll notice with delta, this is typically where you see very deep sleep. So once somebody hits that very first stage of really deep sleep, you see these very prominent, uniform waves. And what's interesting about the delta waves is it does mimic what it looks like if you're in a very deep stage of sleep. And like a good example would be coma. So in a case where the brain is no longer functioning as well, you start to see very uniform waves. So if you were to do an EEG on somebody and you saw these very high amplitude slow patterns, that really could signify brain death at that point in time. You don't see beta waves, you don't see alpha waves of any kind. It's just very rhythmic, slow activity. That means the neurons are just firing like this. And that's all that's happening. You don't get a lot of this sort of chaotic activity. Now, those are the 4 that I'm giving you. The other ones that I want to talk about are just sort of other fun topics. So they do have, one of the things that they were looking at are also called gamma waves. And gamma waves, if beta waves are about 13 to 25 cycles per second, we have gamma waves. And these gamma waves are really fast patterns like this. I mean, I'm talking about really, really low amplitude, high frequency. And where they found these, what's really interesting about this is they found that we get a lot of firing in experiments where they wanted to test what's called the unity of consciousness. And we actually don't have any units. Same one as that, right? Or Tuesday. And what they did was they had monkeys, and they measured these waveforms in monkeys back in 1968, where they put them into basically, I don't know if it was a cage or a room or something like that, and all the monkeys were just literally just sitting there. And they all noticed that they were all together, and what they noticed about that is that they all started to exhibit. When they put the monkeys together, they found these fast gamma waves that were happening. And they thought, why is it only when these monkeys were together that they started experiencing them? And so they started to speculate that it had something to do with an awareness of your consciousness. This is like they speculated it was sort of like that free will, the control to, the ability to control whatever is in front of you, that you're interacting with the environment, that you actually have a place in the world, and that you're not just there just to feed and to reproduce and so forth, that you actually have these interactions. And they did the same thing with Tibetan monks, when they all have when they all start meditating together, they all will just kind of sit there together, and they eventually will find they exhibited the same gamma waves. So that must mean something, that there is something to do with these particular waves. Another thing that they did back in Frankfurt University is to take those same gamma waves. They can basically make those same electrical waveforms, and they place them onto their head, and they it wasn't really more of a shock, but they're basically forcing the electrical activity onto people that were sleeping. And they put it right up on the frontal lobes, and what people experienced when they woke up, they said, I felt awake during that entire time. So they were actually asleep. They knew they were asleep. They had them all hooked up. They can tell they were asleep. But what they experienced was something called lucid dreaming. Has anybody ever heard of lucid dreaming? So lucid dreaming is a form of dreaming, and if you're ever interested in it, just read about it, look up some YouTube videos. It's the ability to sleep, to dream, and then to control your dreams. So you're literally just being able to, it's like being in an interactive movie where you're like, I want this to happen, I want this to happen, I don't like where this is happening, I'm just gonna move away from things. And there's a lot of interesting stuff. If you ever even look up some of the audio, they have things that will try and elicit lucid dreaming, and what they're playing back is a very high frequency sound, and they'll take you through different stages of sleep to elicit those waveforms. Really crazy stuff. I remember playing around with it. I got freaked out. I'm like, oh, I don't know about this, because I really did feel like I slept. Like when I woke up, I was like, I need to go back to sleep. I need regular sleep. This was too much. I don't really remember what happened, but nonetheless, some scary stuff. Maybe something that my fragile mind couldn't handle. So with that, those were gamma waves. Another thing that they also looked at were another type of wave that's very similar to beta waves. But where did they find these beta? They're called mu waves, MU. These mu waves they found to be specifically in the primary motor cortex. Again, the most posterior portion of the frontal lobe, the area that corresponds to motor. And they measured a lot of these mu waves, and it all corresponded to motion. So let me take you back to an example that I mentioned earlier, tying in this example that I had in college. They had me, if you remember my story from early, our second lecture, I remember I mentioned I was inside a PET scan, and they had a video, and they made me watch this video of someone putting the coffee cup back, picking it up, putting it back down. And what they were looking for specifically, and they injected me with contrast, they wanted to find an area of the brain that corresponded to the movement of the arm and the hand, but specifically with the left hand. And every time I kept thinking about it, it would start to get lit up in this scan. So they do the PET scan. They also did a functional MRI where they did inject me with contrast to find that particular area that corresponds to the action of picking something up. And they wanted me to not focus on anything at all. So that way they're able to find, okay, here's the primary motor cortex. This is the area that corresponds to the hand. They've been able to identify that area because that area is metabolically active. Marlon is thinking about this over and over and over again. This must be the area. So what they wanted to do to me next was then inject me with this, they had that helmet on, they had this probe, and they wanted to shock that area. And that's the part I didn't do. But what's interesting about these mu waves, if they measured it, if they were able to measure mu waves, they noticed that that area of the brain when I'm not thinking about, well, maybe not me in this case, but in the studies that they've done, if they measure those mu waves happen all the time. But the second you start thinking about emotion, or when you start experiencing that emotion, it turns the mu wave off. So in other words, mu waves are turned off when you think or do something with your muscles. Okay? And when they're able to do that, and they have that area mapped of the primary motor cortex, they figured, well, if we can receive the signal from the brain, does it mean that that's what's used to control that particular body part? So with that, what they wanted to do is receive those messages, and if they could receive those messages from the brain, could that directly signal something related to motion? So could they predict someone's motion by just reading the brainwaves taking place? And so this gives rise to the whole mind-machine interface. So to give you an idea of what they are trying to do, is if they read those mu signals from the brain, from the primary motor cortex, and they can send that over to something else, not connected physically to me, send those same signals over to the motor neurons that are responsible for moving that motion, could they get me to sort of think about a motion and get some other external thing to mimic that motion? So this ventures into the whole, what's called MMI, mind-machine interface. It's the ability to receive signals from the motor cortex, the areas that they want to read, and detect that movement pattern and be able to use that on something external. So this gives a lot of rise into telepathy, the ability to control things with your mind, specifically with motion. And that might be a little far-fetched. I know we're sticking strictly with physiology, not trying to venture off into far away from reality. But the reality is, there's a lot of interest in this. Okay? A lot of military interest in our ability to control things with our mind. Things with space exploration. If we can start to control robots just through the virtue of thinking about things, being able to navigate, and so they've done a lot of experiments of rat to rat. Duke University was able to take a rat and elicit a tail wagging in one rat, and they had it plugged into a bunch of EEGs, and they transmitted that signal over to another rat where they severed the spine. And they severed the spine obviously from the brain to the tail, and they wanted to see if rat number one could do the tail wagging, and connected it to the muscles of another tail, could that elicit the tail wagging on another? And they found they were able to do that. So naturally they thought, okay, well, let me try something else. Let's try humans and rats. So they had a guy watch a TV screen, and I think I have a video for that. Yeah. Let me just show you the video. How about that? So this guy is going to be staring at a machine, and what they're going to be doing is every time he sees a flinch, they want to see, can we get this same rat who has its spine severed, could they also get this tail to wag as well? So here you can see him watching. He's literally just watching the signal. He's got his EEG on. And every time what we're looking at here you can see the tail starting to move. And every time we reach this portion where there's a little spike, that causes movement in the tail. And that's all through him thinking about when that's taking place. I don't think it happens again, does it? But you get the idea. One more time at least, come on. There it is. So these are the things that we think, okay, maybe there must be some way to do it. Another thing they were able to do so now they had that, and another thing they wanted to do was also human to human. So they used a finger twitch. So they connected two people together, and this was over in Washington University, where they had these two people completely in different rooms, and this would send a signal. He was I think watching the same type of flashing sign or something like that, and whenever he got a signal, he had to think about the motion of twitching that finger. And it came across, and what they were trying to do is the same type of experiment where they could cause a probe to see, will he receive that signal? Is there some matching? Would he know if they can sense that mu signal, would he know that he had twitched? And so they were, I don't remember the results of that, but it didn't seem like that cool of an experiment. What was a cool experiment was this. So here they take a monkey, okay? And what you notice about the monkey, they got the machinery up on here. They have the monkey's head resected, and they have the brain kind of exposed. And one of the things they wanted to do was find the primary motor cortex. And they have electrodes plugged into the primary motor cortex, and they have him connected to this machine. And this machine is an external arm. Obviously, the monkey can't really move its hands or anything like that, but it over time has to learn, and they do a lot of, keep in mind, they can't just plug you with into a machine and all of a sudden you learn how to control this thing. This took a lot of time to learn the motions of moving things. So he learned eventually how to manipulate this arm all through his brain by itself. So by manipulating this machine, you'll be able to see how he's able to grab the treat itself. You see the wires back there connected, and of course they don't show you what's happening up there. So there's a lot of implications behind stuff like this. If plugged into a machine and reading these same mu waves, and those mu waves corresponding to muscle motion and movement, could we get people that actually lose the capacity, people that are paraplegic, people that are quadriplegics, could we potentially get them to be able to move external machines as well? And we'll see that on our next slide. Okay. So this lady, I'm going to skip a little bit of the video because they get into...
I always said, except for this walking thing,
it's 9 minutes. I know we kind of want to just cut to the heart of this whole story. But she's a quadriplegic that injured herself during... she injured herself, I forget what type of injury or what accident happened. And she signs up for this experimental protocol. She has absolutely no control of her arms and legs. And so what they wanted to do with her is part of this experimental protocol. This is a research protocol where they would install these electrodes into the primary motor cortex. And keep in mind, there's nothing wrong with the motor cortex. That's still functioning. It's just the motor cortex that leads up to the extremities is severed. But she'll still think about it. She can still think about moving. So they want to get these probes to connect into the primary motor cortex to see if she can learn to manipulate this machine that's supposed to basically be the human hand. And so these machines have, and this goes into a little bit more of the technical side of the machine. I really want to get to the surgery itself.
So that you can record the responses of individual neurons as well as...
So right now she's going through the surgery to have the implementations.
And so in this case we implanted two. These are implanted along the motor strip and really tried to target this so that we would place one centered in the areas that were activated when she was imagining using her hand, and then place the second one in the area that was activated when she imagined using her shoulder. 2 days after surgery, we went over to the rehab hospital. We took the system with us so we could plug her in. And we were just curious to find out whether or not we could record anything that sounded like a neuron.
Okay, well, let's turn my phone again.
When we hooked her up for the first time and we had her think about moving her little finger, right then we knew we'd done everything really well and that we had a really good circumstance so that, you know, we could do good recordings and that Jan was going to work out really well. Now go ahead and try moving your wrist. Oh, there we go.
Every day as we get closer, as we drive closer and then we come up the road.
So these are the electrodes that they use to plug into the machine, and the machine would be controlled to that robotic arm.
What's going to be new? And every time we do something new, it's challenging at first, and then when I get 28 out of 30 or whatever the score is, and they say that was all you, that wasn't the computer doing it, that was all you, I just can't stop smiling. It's so cool. I'm moving things. I have not moved things for about 10 years.
Yay.
All right.
There we go.
Very fast.
Initially, we started out with her doing three-dimensional control of the hand. And so she's controlling the endpoint position of the arm, moving forward, backward, up, down, left and right, that kind of thing. And so since she was making good progress with that, we decided to try to add in orientation of the wrist, so rotating in all three directions, and then also adding in grasp. So that's where we're sort of focusing now on 7-dimensional control of this endpoint position, orientation and grasp, and sort of really working on interacting with objects and being able to move them around her environment.
This counterclockwise.
I used to have to think up, clockwise, down, forward, back. Now I just look at the target and Hector goes there. It's not a matter of thinking which direction anymore. It's just, I want to do that, and my brain knows what things to move to make that happen.
Right now, all Jan is doing is imagining moving the arm exactly where she wants the arm to go, just like you or I would control our own arm. What we've done, interestingly, with Jan is that normally the computer controls a little bit and Jan controls a little bit. At least that's how we started. And Jan didn't want to know when we switched the computer control off. She didn't want to know when we weren't assisting her in the task. And almost as soon as she said that, we flipped all the controls off. So we gave her complete control of the arm, and then she controlled it perfectly.
Yay!
That was incredible.
20 out of 20.
You made that look like training.
I'm performing the computer because it doesn't usually do more than they do.
There are things I regret not doing when I was able, not skydiving, but some adventures I would love to have had, and I regret not doing them now. And this is the ride of my life. I keep saying this is the roller coaster. This is the skydiving. It's just fabulous, and I'm enjoying every second of it.
From the beginning, we've been really focused on, how much control can you get of this arm, moving the hand and wrist and everything. But, Jan's goal has really been to feed herself a piece of chocolate.
Oh, I wasn't paying attention.
There we go. Just took a little time.
One small nibble for a woman, one giant bite for DCR.
I think the highlights of the study are watching Jan feed herself, watching Jan move objects, watching her complete tests that we normally have for people with stroke who are trying to recover function, and having this beautiful fluid control of a robotic arm through thought.
I think it would be great to really get her to the point where she can actually feed enough food that she can satisfy herself and that it becomes routine. I guess I'm a little worried that she's eating just chocolate and cheese. We need to start to vary the diet a little bit.
Today I accomplished a goal that I set before the surgery even took place. They were asking if there was something special I wanted to do, like touch my children's cheek or hold my husband's hand, and I said, my goal is to feed myself chocolate. And I did that today. I fed myself chocolate, and then string cheese, and then a red pepper. So we're trying to get a balanced diet here.
You watch Jan smile after completing a task in a time that's close to what I could complete that task in, and you think, wow, how do we just have to take the next steps where we can make this a clinical tool, not a research one.
It overwhelms me. I'm so glad I did this, and I know some people will watch this down the road and say, you know what? I could do that. I can't hold a job anymore. I can't even talk to my children or pick up a pen to write. But I can do that, and I can make a difference for people in the future. So I'm excited to think that our work here in the lab will inspire more people to come forward and for the work to continue.
So that's the story. They've had, and I really wanted to grab other videos too, because not only have they done this with hands, but they've also been able to do it with walking apparatuses. So you'll notice that when they do get them plugged into machines, there's this whole learning process, just like you saw Jane with the hand. It took quite some time for her to, as she stated herself, she had to think about the motions first. She had to think about moving the hand, turning it clockwise, moving it forward, and then eventually, after a while, she started to learn to just think about, not even thinking about the motion, just thinking about where she wanted to move that hand. And she said, eventually, it just started to do it on its own. So that process is just exactly what babies do when they first. If you ever watch babies when they're super young and they start to pick up items, they do exactly that. They start to stare at the object, they look at their hands, they move things, and eventually after a while they get to learn how motion works. They can perform motor functions fairly easily. They don't think about having to pick something up, they just do it. That's the same thing that's happening with Jane. And they've been able to do it. This clip that I was talking about, they've been able to do it with assisted walking machines. So they have these people that are paralyzed, that lose the ability to walk, and they have them fitted with these machines. And from what I saw last time, they're fairly rudimentary. It's like a big machine with these 2 robot legs. And they do the same thing. They get them connected to the primary motor cortex, and they have that person learn to eventually be able to move. And obviously that's going to be more of a longer task, a longer arduous process, because there's a lot that is involved in motion. They learn how to be able to balance themselves and be able to mimic certain motions. And for the Super Bowl, one of the kickoffs, and I can't remember which one because I'm not really that into sports, but one of the kickoff for one of the Super Bowls, they had a guy kick the ball, and you saw him on this robotic machine. That robotic machine was exactly the same MMI interface that they had doing. And people weren't really all that aware of what that was. They just saw some dude with the robot legs that came up and kicked, but they didn't really understand the significance of it. It was a man that had absolutely no motor control of his legs, regaining that ability to be able to move himself. I haven't watched. I remember I don't really watch the Super Bowl. I probably watch commercials, and I know when that portion happens, I kind of tuned out. So I didn't really pick up on it. I've been wanting to find more of the clips of it. I just haven't gotten around to it. But nonetheless, what we're really trying to move towards is this. This is where we start to see a lot of military influence, a lot of military funding for this type of stuff. This is a project that was started in 2012, where they have a man in a cockpit, but there's no actual motor controls. And so they put him in this basically a simulator to see if he can move the fighter jet just simply by thinking about the motions, and without the need of a control or anything like that. What they're trying to move towards are just unmanned machines. You can get people that are connected to basically a terminal, just like a big video game. They just have them thinking about the motion, and then you get these fighter jets moving around. This was a fairly big project, but again, kind of a shut down project just because of the implications behind things like this. Because obviously, you're starting to bridge machine. I mean, obviously, fighter jets that are big, obviously very expensive weaponry connected to mines and stuff like that. So there's a lot of suppression with stuff like that. Kind of similar to what they're doing with AI. I know we've made some huge strides in artificial intelligence, and yet then they're saying, I don't know about artificial intelligence yet because we don't want these machines to think on their own. So with that, there's a lot that goes into it, but then again, a lot of questions about ethics of these types of studies. So that's it with our mind-machine interface. The next thing I want to get into is something a little bit more applicable. I don't know if any of us are really going to be doing any mind-machine interfacing stuff. If you ever do, that's really cool stuff. Let me know what you're doing with it. But let's get to something that we all love. Okay? And what I want to start talking about is the whole concept of sleep. And what we talked about in our last lecture, we talked about the reticular activating system. It's the RAS system. And you guys remember my story about the whole caffeine nap? Mm-hmm. Yeah. So with consciousness, with awakeness, what's happening is we get all this sensory information that travels to the reticular activating system. And that system is a series of brain structures that are all related to receiving sensory information, and it's all tied into wakefulness. And as we go through the course of the day, we get all these neurotransmitters that are blasted through the RAS system, and the receptors are constantly bombarded by these neurotransmitters. And eventually, over time, those things start to fatigue, the RAS system starts to fatigue, and that's where you start to get the feeling of tiredness. And that's where I mentioned in our caffeine nap, that if you try and drink caffeine when those neurons have been constantly bombarded by neurotransmitter, it doesn't really do very much. And so that's when I talked about taking a nap. because then it'll reset the RAS system, and then you can start utilizing caffeine to stimulate those areas of the brain. Now, one of the things that we're going to learn later on is melatonin is a hormone that's secreted by the pineal gland. You might have heard of melatonin as one of the things that is involved in circadian rhythms, specifically related to sleep. You've probably seen derivatives of melatonin being sold in health stores to try and elicit sleep. But melatonin is specifically used to inhibit the RAS system and therefore trigger sleep. So one of the things that you're going to see when we do get into sleep is the different phases. I first want to talk about two different phases that are characterized during our electroencephalograph. The two types that we're going to be able to see are REM sleep, which is rapid eye movement, and the other being non-REM sleep. And this is what you'll be able to see later is our deep rest. And if you were to take a look at what happens during rapid eye movement is dreams occur. Has anybody ever watched anybody sleep? I know it sounds kind of creepy, but you ever watch anybody sleep, or even dogs, for instance? You watch them and you see eyes moving around quite a bit. You see a lot of muscle twitching going on. What's going on is there's a lot of dreaming. If anybody watches somebody sleep, you know they're dreaming fairly heavily. So there's a lot of activity going on mentally. So what you would expect to see are like beta waves, very similar to wakefulness. A lot of brain activity happening. So we would typically see something that looks like this, a lot of wakefulness. And what's happening in REM sleep, again, low amplitude, high frequency oscillations. We know that REM sleep is good for memory storing. Why do we need to dream? A lot of theories on dreams and things I won't really get into because that's more so in psychology, but memory storing. Learning is a big portion of it. And then mood regulation is also something important. So these are things that are fairly prominent, and I've always been really interested in learning more about dreams and what they really specifically mean, because sometimes dreams seem to trigger some distant memories or sometimes fears or anxieties. Sometimes, just how varied they are in nature. I don't know where they come from. I really wish I'd studied more about dreams, but nonetheless, here we are in physiology. Non-REM sleep, on the other hand, is very high amplitude. So again, these types of waves, high amplitude, low frequency waves. Again, these are more your theta waves. These are the deep sleep waves. And typically when you look at these, you'll see these long delta waves. And what you'll also find amongst these delta waves are what are called sleep spindles. And so these sleep spindles are these little bursts of high frequency amplitude. And what you would typically see with deep sleep, somebody not really moving much, but they'll have just at some point they might just twitch a little bit, and you can see it. And if you were to measure the EEG when these sleep spindles occur, they'll see a little twitch. So watch somebody in that first bit of sleep, and you watch them twitch, and they're in deep sleep. There's nothing you can do to wake them up. Have somebody in REM sleep; they might be dreaming. You just kind of wake them up a little bit, or you can just like touch them, and they'll just wake up. Why? I'll show you in just a little bit. So these sleep spindles, people will just kind of jerk, and that's it. And then they're back to this deep sleep. There's some theories behind it. You see children especially that have these little sleep bundles, or the sleep spindles. And it's theorized that it's all due to branching the neural connections from the primary motor cortex to those areas of the brain. We're trying to strengthen the synapses. Again, not a lot of foundation for that type of information, a lot of theories behind it, so I won't really get too much into it. Now let's look at the different stages of sleep. And here you can see the stages of sleep from 0 to the 8 hours you're supposed to be getting. Okay? You can see the different sleep stages where we go from awake down to stage 1, 2, 3, and 4. And each of these 4 stages are characterized. Stage 1 is a light sleep. Stage 2 is more of the sleep spindles. Here you get to a little bit of a deeper sleep. Stage 3, you start to see a lot more of the delta waves, even deeper sleep. And then stage 4 are mostly our delta waves. Typically where you would see this would be the deepest stage of sleep. This is what non-REM sleep, someone who is not responsive. You can sit there, you can slap them awake, you can shout at them. I know in college I've had people, like, I remember somebody held a trumpet to my ear and tried to wake me up. I couldn't wake up. When I did wake up eventually, I did hear ringing for quite some time. But that is our deepest stage of sleep. And what I want to just show you is how we characterize those stages of sleep. What's happening throughout those 8 hours. And what you'll notice in the first hour is we go from awake down to that first stage of sleep. Okay. And what happens during that first stage of sleep? Does anybody want to like share what happens when you first fall asleep? We're like doing weird things. What's that?
Easily awakened.
Really easily awakened. What's something else that happens when you sleep? Moody? Well, no, I mean you're actually like falling asleep, like that brink of falling asleep. Like you're like laying there in bed, and then you're just starting to like pass out. You get drowsy. Okay. Yeah. Does anybody ever like twitch a little bit?
Yeah.
You twitch. You have that feeling of falling. Yeah, you get those. Those are called hypnic myoclonia, and it's that feeling of falling. It's characteristic. And when I said that, everybody said, "Oh yeah, I get that feeling." Yeah, it's always something. It's either a dream where I'm like on a roller coaster and I'm going down, or sometimes it's a car crash and I'll just wake up and I'm like, "What the hell is that?" And then you'll fall back asleep.
I thought of that when you said sleep spindle.
Like the twitching?
Yeah, because I was like, that made me think of falling in my dream.
Yeah,
Is that big twitch?
that's deep sleep. This is actually within the first like second when you're actually sleeping.
So then is it a sleep spindle when you get that feeling in the middle of your sleep?
Quite possibly. What might be happening is you come right back to awake and then bring it back down to stage 1. This is typically happening if something like something interrupts your sleep. You're conscious, you're awake, and then you fall back asleep, and then you'll just kind of get that
Like
twitching.
an earthquake? Like an earthquake?
Well, an earthquake's an earthquake. That's a little bit different. That'll wake you up no matter what it is. But certainly, you can have like a dream of an earthquake and you just wake up immediately. And this is happening right when you fall asleep, and you might look at the clock and be like, I didn't even sleep yet, what happened? But typically and I want to take that back, because typically with hypnic myoclonia, you twitch, you fall back, and you open your eyes, and then you just fall back asleep again, in most cases. What's also interesting about this first stage of sleep, I don't know if any of you guys have ever like early love, like you call somebody, you talk to them late at night, and then you're like sitting there talking until the point that you fall asleep. This has happened maybe once on me. I'd wake up like in the middle of the night and be like, wasn't I talking to such and such? I don't remember that. So what happens right before somebody sleeps is that for the first couple minutes, like right at the point when you reach that point of unconsciousness, you actually have a tendency to kind of forget what happened right before. So you could be talking about something, you could be thinking about something, and then when you start to drift, you forget what exactly happened. This kind of explains the whole reason. How many people here snooze in the morning? How much time do you set your alarm to snooze?
Intervals of 5 minutes for like an hour.
5 minutes, 10 minutes? So for those that snooze for about 10, 15 minutes, okay, how many was it, 10 minutes? For you guys that snooze for about 10 minutes, do you know how many times your alarm has gone off? No? Okay, that's a feed soon. Generally speaking, people that for about 10 to 15 minutes, all right, that's a second alarm, I gotta get up. All right, that's a third alarm, I gotta get up. If you set your snooze to about 3, 4, 5 minutes, you don't really remember that stuff, because like I said, when you fall back asleep, you forget what happened a couple of minutes ago. So it's not uncommon when you look at any sort of like timers, clocks, you look at apps, they always set it to 10 minutes. Why don't they ever set it to 5 minutes? It's ineffective. So if you want to snooze for about 10 to 15 minutes. It's a lot more productive in ways. Until you keep snoozing to the point that you turn off your alarm and then you wake up at 9 o'clock. So That's something really interesting about bringing that bit up and just kind of tie in that the whole thing I was talking about earlier when you're talking to someone late at night. I don't remember what I talked about like a couple minutes before. It's actually interesting because at that point when you know somebody's super sleepy, you might be able to get a little bit more information out of them because they won't remember it the next day. All right, so now we're venturing off. We're venturing from awake down to the first stage of sleep. And what you notice, take a look at where you spend the most time in deep sleep. And deep sleep being the deeper stage, stage 4. Out of this entire map of 8 hours, where do you see the spending most of your deep sleep in? I think around 8 hours. Around 8 hours, you're more towards the higher, deeper stages. Where do you see the deeper stages of sleep? Stage 4. Within the first hour, right? That first hour, you spend that most time. So watch somebody fall asleep and give them about 15 minutes. There's no chance of waking them up at that point. They're done. They're at deep sleep. They reach that fast portion of that deep sleep. And that deep sleep is fairly important because we're able to turn off our neurons at that point where you would suppress any movement of any kind. There's a lot that goes into about that deep sleep. But what you notice after coming back after that first hour, you'll note, okay, now we're starting to climb back up into the lesser stages of sleep. And then you hit these stages of REM sleep, where REM sleep, you notice REM, rapid eye movement, this is the area where you're dreaming, is very similar to a wakefulness. So you get these bursts of REM sleep, and then you go back into deeper sleep, and then not until the third hour you see the REM sleep. Certainly, you hit different areas of REM sleep.
Is it better to wake up during REM sleep?
It's better to sleep all 8 hours. It's really
yeah, but I mean, if you don't have a choice.
Which actually is a good question. On average, how many people here get less than 3 hours of sleep a night? No? Nobody? Okay, good. How many of you guys get under 5 hours? Really? Oh, you can sleep that well?
Sometimes.
Okay.
Most of the time.
Most of the time? All right. So how about 6 to 8 hours? Most everybody else. Okay, so that's everybody else. So one thing you might notice about sleep, take a look. I used to sleep about like 4 or 5 hours, and I was definitely a little crankier person. But nonetheless, if you were to take a look at where would you see somebody spending the most time in REM sleep? Is it the first half of sleep, or is it the second half?
The second half.
The second half. So it's better. You see the reason as to why when we start, especially you start, and I don't know if you sleep at, let's say, probably not 10 o'clock tonight, if you're not already asleep now. If you go to bed and you get to about the 6th hour of sleep, maybe about 4, 5, 6, 7 o'clock, whatever time you sleep at, that's when you get the most number of dreams happening because the latter half of sleep, you spend more time in REM sleep. Okay? And the times that you would wake up, you're most likely to wake up towards that very end where you're already at REM sleep. And you wake up, you're like, oh, that was a crazy dream. You remember it. Okay? You might not remember your latter dreams that that were taking place in REM sleep. So it's crucial to see that people that really lack sleep, that sleep less than 5 hours per night, get comparatively less REM sleep. And what I mentioned about REM sleep, notice what I mentioned, memory storing, learning, mood regulation. What happens with people that are not getting enough sleep? They're moody, extremely moody. They forget things. For some reason, I don't remember that. Forgetfulness. And typically have difficulty kind of picking up, I wouldn't say learning, but learn at a little bit slower rate. And that's due to that lack of sleep that's taking place. So it's imperative that people really get the 6 to 8 hours in order to get that. They have done these types of sleep where they would get people sleeping. They'd look at where these REM sleep segments are taking place, and they would have like a little shock to wake them up, and they would deprive them of REM sleep. And that's where they started to find functionally they were impaired. They had inability to recall memory. They had bad short-term memory. They mood extremely moody, and so forth. So these are, like I said, the different stages of sleep. You would see deep stage of sleep happening at the first 2 hours, REM sleep taking place at the very end. And I'll talk more about the implications of why we need REM sleep a little bit later on. All right, so language. So these areas of the brain are involved in language. And so we have two specific areas. Broca's area, which involves articulation of speech. This is the ability to speak, how clearly you can speak. And what you would notice about people that have damage to this area have difficulty speaking. They can't typically say things, and they're called aphasias. They mumble, they stutter a lot. Yes? What's that?
I was gonna say, oh, is that like stuttering?
Stuttering, yeah, exactly. It's just the inability to actually speak. They know what they're trying to say, they just can't formulate the words to say what they want to say. It doesn't affect their ability to understand. So you can talk to that person, they understand exactly what they're saying, they just can't communicate it back. Wernicke's area is the other side of it. Wernicke's area is involving the comprehension of language. They can speak fine, right? But they can't really understand what you're trying to say. So speak to them, and they'll mutter off about something else, and it'll just be a string of nonsensical words. So people will typically, with Wernicke's area, they might just start speaking things that make no sense at all, because they can't comprehend. They haven't been able to comprehend anything that people have been saying. They know the words, it just doesn't make sense of what they're saying. So these are the typical aphasias. So aphasias are things where you start to see damage in those areas. And I have very quick examples that I can show you of Broca's aphasia, where, again, able to understand the question, but not able to articulate it.
Now tell me what this thing was with your legs last week or week before.
No good.
Did you expect? Couldn't really say it. He knows exactly what he's trying to say.
And
So again, he doesn't say anything at all. Now let's take a look at Wernicke's aphasia. So this lady, the interviewer will ask her a question, but you'll see, she won't be able to, I won't spoil it, let me just get right into it.
Thanks. And say, baseball player. Caterpillar, cantaloupe. Okay. All right, I'd like for you to do some things now with your mouth. First thing I'd like for you to do is stick out your tongue. Stick your tongue? Show me how you would.
You can see that she has some issues, obviously being able, nothing really wrong with her ability to speak, but really can't understand very well. And so typically you would see these aphasias as a result of damage to those areas of the brain, such as stroke, or any traumatic injury to that particular area. Another area is the angular gyrus, and this is actually in the parietal lobe, and this is all related to memory retrieval, so things that are responsible for the integration of auditory, visual, and somatosthetic information. This is memory, this is perception, and is related also to memory and taking numbers and being able to process them. What they find about people that have damage to these areas is that you typically find dyslexia, so the inability to be able to see the patterns in words, inability to do math, sometimes see issues with attention and so forth. What they've done before is the stimulation of these areas, and what they know is by stimulating that area, and literally like probing into that area and shocking that area, is if this is an area that's responsible for perception, people have found that they've had what are called like out-of-body experiences. Something where they feel like they're, and I don't know if you've ever seen people that have like near-death experiences, and they feel that they've like stepped themselves away from their bodies and they can watch everything that's going on. So it's like this feeling of detachment from receiving sensory information. The last thing on here is the arcuate fasciculus, and this is basically all related to information traveling between the Broca's area and the Wernicke's area. Basically, when you look at all of speech, the whole concept of language, it's not really just the ability to speak, but it's also the ability to comprehend things. So obviously you're going to have information traveling between these two areas. So a lot of these pathways that we mentioned way earlier, if I can get back to it. But these are going to be your association tracts and commissural tracts. Basically what you're doing is sending information back and forth to be able to communicate to each other. So you take in information, sensory information goes from one area and then travels as you're sitting there communicating and understanding the words that are coming out, putting together the words that you need to speak will need to travel back and forth to different areas of the brain. And of course, if you're trying to speak, you get to the Broca's area, and then you're going to have to take that back over to the primary motor cortex, because those are the areas that are going to control the lips, the mouth, the tongue, to be able to speak and formulate the words. The last thing I want to leave you before we go on break is emotion and motivation. So these areas, this is the limbic system. The limbic system, again, not a specific structure, but it's a whole collection of all these nuclei that are found in this whole ring around the midbrain. And these are all the different things that you would see. Hypothalamus, hippocampus, amygdala, the thalamus, which is receiving sensory information, the corpus callosum, which is sending information between the left and right hemispheres. And then, also notice on here, the olfactory bulb and olfactory tract. And olfactory is related to the sense of smell. So looking at the emotion and motivation, these are all things that are related to the states of emotion. It's all involved in these systems. It drives emotion, it's heavily tied into memory. So if you ever think about situations that evoke memory, typically are tied into things that were emotional. What's also interesting, and then we'll learn this in our sensory lecture, is olfactory, which is our sense of smell. You'll learn that that sensory pathway leads directly into the limbic system. So if you ever to think about certain memories that you ever had, you would notice that the thing that you might remember the most vividly is the sense of smell. Remember in anatomy, if you were kind of averse to dissection, you remember the smell of formaldehyde. So if I were able to release, if I cracked a little bit of formaldehyde, you would sense it. You would think, oh, I'm thinking of dissection lab right now. And probably the emotion that's tied into it.
Even with like you smell like something that your mother cooked when you were
Certainly, there's a certain emotion that's tied to that. The certain, maybe the certain perfume or cologne that somebody used to wear, you might smell it, you might think of that person. And it's actually fairly subconscious. Before you're even really consciously aware, that memory is certainly triggered. So these are all the things that you were sharing with me are things that are tied in. And you'll learn that the olfactory sense skips some of the pathways that we're going to be able to see in our sensory lecture. And it's all tied into that. So what exactly is memory? And we know that it's divided with short-term memory and long-term memory. But what we really need to get into is how we take that short-term memory. How do we take information that we just stored and integrate that into long-term memory? So what this is, to give you an idea of what we're thinking about here, is the information that you're learning, the stuff that you're learning about physiology. One thing that you ever learn about integrating something into long-term memory is repeating it over and over and over again, right? So there's things that you remember best. Like there's certain people that will, they can read information, they can quickly remember that information. They've got a great short-term memory, but ask them a year later, I don't remember what that was about. Then you get certain people that, especially through repetition, maybe they'll study and they got to study over and over and over again. They got a great long-term memory and they'll remember it for the longest time. So these are all different things, different ways we store memory, but really what we want to get into the mechanism behind it. And again, this is very brief generalization into it. A lot more we can get into the physiology of memory, but given that this is an intro class, we don't spend too much time into it. But short-term memory is basically the things that happened recently, recent events. This is looking at a phone number and saying, I got to remember these 10 digits, and I got to remember for the next, 10, 20, 30 seconds. And then after that, if it wasn't significant, then it's removed from your memory, and that's really it. Now, the whole concept of taking that short-term memory and integrating it into long-term memory is some concepts that we'll talk about in the next slide over. But the areas that are extremely important are the hippocampus. The hippocampus is the area that's very critical to memory itself. It's how we take information. When we look at short-term memory, we take information that we just learned, anything that we deem relevant, and store it utilizing the hippocampus into areas like the medial temporal lobe. And with long-term memory, with our short-term memory for the most part is finite, meaning we can pick up a certain amount of short-term memory, we can remember it. But long-term is indefinite, meaning once you remember it till the day you stop existing. These are things that, barring any traumatic injury or any stroke or anything like that, you'll remember this for the rest of your life. Now, when you look at long-term memory, we take short-term memory, and memory consolidation is the process of taking that short-term memory into long-term memory. So if you've ever thought about how do I store things, right? We know how computers store information, but how does our brain store information? You might have probably heard at some point, oh yeah, you're building more brain cells. Technically, no. You're not really doing anything with the brain cells. If you hear the term, you lose brain cells and you never get them back, yeah, okay, sure, that's true. But your brain cells aren't the ones that are fully involved. Do you guys remember the other types of cells that were involved in the brain? I'll give you a clue. It had something to do with some of the questions I asked you in your quiz. There were neurons. There's also another type of cell. It starts with neuroglia. Neuroglia. So we've got tons of neuroglia that are involved. They're the supporting cast. And in fact, even though the neurons might be finite, meaning we have a set number of neurons, there's a lot of communication that happens with one neuron to the next. In fact, there's ways of enhancing that communication, which we'll get into on the next slide. But really what it comes down to is when we store memories, it all comes down to the activation of genes. This is the DNA that contains specific areas. We activate those areas and then we lead to protein synthesis. We're actually those memories form these proteins, and those proteins are involved in enhancing the connections between neurons. So when you're learning things, you're actually enhancing the connection between certain areas of the brain. You're actually forming new connections. So while the number of neurons stay the same, we might start to build new axons that form off existing axons. Maybe we're able to route things around a little bit faster. Maybe we're myelinating axons to travel faster. What's interesting about babies, right? So newborn babies actually don't have as many myelinated axons as adults. So when you look at babies, they really don't do much. They have reflexes that are innate, reflexes that we'll talk about, but they can't really move things. They can't even really focus their eyes to look at people. So you get a newborn baby looking at you, and it's kind of unfocused. It's really creepy. But as babies start to learn to be able to focus their eyes, they have to first kind of think about it and do it, and then eventually they just start doing it. Just like we showed you with the whole video of Jane, where she had the concept of taking that arm, and eventually she just learned how to do it. So what's happening in that process of learning, especially with newborn babies, is that we start to myelinate those axons. Remember what we learned about myelinated axons. Myelinated axons is a form of saltatory conduction. It's a way of speeding up the information, speeding up that action potential traveling through the neuron. So what you start to notice is that we can have information travel faster. We can have these areas of the brain, the association tracts, the projection tracts, all of those start to enhance, and we can speed up the connections between certain areas. And this is all through that protein synthesis, where we can start to bridge those connections between neurons a lot better. And what types of things will we talk about? We'll talk about that on the next slide for long-term potentiation. Things that you'll also see about the cerebral cortex, areas of the brain. Not only did we see regional specialization, you'll see the same thing with memories. So visual memories, we have certain areas that are called visual association areas. So memories that of a visual nature, if you like close your eyes and you can picture something happening, those are typically happening back in the occipital lobe. So these are memories. When we look at learning, for instance, learning how to walk, learning how to ride a bike, for instance, A lot of those areas are going to be stored in areas nearby the primary motor cortex, areas that we store that information. Prefrontal lobes, typically things, the frontal lobe is right there in the very front, things that we saw in the lobotomy, that were excised during the lobotomy, typically with mathematical calculations, things that are analytical in nature. So like I said, these are things that are involving the hippocampus. There's a story that I have of a kid back in 1953, a kid named Henry Molaison. What happened with him is that he had a bike accident at the age of 7. And this kid, when he hit his head really hard on the ground, and he started having seizures. And these seizures were so profound, and they kept, they were doing some of their earlier scans, and they were finding that areas that were hugely excited were the hippocampus area. So they went through this procedure just to resect that area. If they can sever the connections by the hippocampus, then maybe they can stop those seizures. And surely enough, it actually stopped the seizures. But remember what I said, hippocampus is involved in memory. So what happened with this kid at the age of 7 is that all of a sudden he had difficulty remembering. He started having difficulty remembering things after the incident, after the brain surgery. So even though he was having epileptic seizures, he couldn't really remember things after the fact. So with the inability to formulate new long-term memories, this is a condition called anterograde amnesia. They can't form anything that happens currently, they can't remember. But anything that happened prior to the incident, they remember really well. So what happened with that is his brain is actually stored over at UCSD for study because it's one of the first cases. Now, obviously, it's still not studying that brain anymore, but it was one of the first cases of anterograde amnesia. Is anybody? Yeah.
I have a question. In that case, would he be able to go to school since he can't learn anything new?
Can't learn anything new. So probably be able to learn things during the day, but that his short-term memory would work fine, but the inability to consolidate that memory into long-term memory is gone.
So he stayed with like a 7-year-old.
What's that?
So anything he learned before, right? So
Everything you learned before is fine. What was that?
It's like the movie 50 First Dates.
50 First Dates? I didn't watch that one. Adam Sandler? That's why I didn't watch it. It has Adam Sandler.
For her every morning.
Yeah, it's so cute. And then she had a baby. She's like, what is this? Yeah, watch the video.
And these were things after the incident, right?
Yeah.
After the incident.
There's also a movie, Memento,
Oh, yeah.
where a person couldn't create new memories and he would tattoo all the things that he found. He would fall asleep and wake up and not remember a thing. Those are things too. Those are anterograde amnesia. Retrograde amnesia is the exact opposite. So these are things prior to a traumatic incident where they don't remember things from their childhood, but they can form long-term memories, which is a more common type of amnesia. Obviously, things with anterograde amnesia, I don't know how functional you're going to be if you can't remember things that happen in the present. So with that, let's take a look at the cellular mechanism. Okay, and this whole concept is what's called long-term potentiation. Everything that I've taught you guys about neurons communicating through the form of an action potential traveling down the axon to the axon terminal, and the axon terminal that we have here, this is the presynaptic neuron, filled with neurotransmitters in these vesicles. These neurotransmitters are exocytosed into the synaptic cleft, and then those neurotransmitters bind to the receptors located on the postsynaptic neuron. Those receptors bind, the neurotransmitter binds to the receptors, and that allows our ions to flow through. If sodium flows through, right, what does that do for the postsynaptic memory? Does it excite it, or does it make it less sensitive? So they excite it, so it's an excitatory postsynaptic potential, EPSP. If you were to see the other ones, here's another one that we're looking at, and you don't have to know this, but there's NMDA receptor and allows calcium into. I did mention that calcium is also heavily involved, but this particular receptor, when bound to, so the neurotransmitter binds to, you'll see calcium flows in. And you don't need to know the mechanism behind it, but calcium comes in and it starts to activate other proteins involved, which are called kinases. Kinases are things that will activate other areas of the genes that will start producing proteins. Remember what I said earlier. consolidation of short-term memory into long-term memory involves the activation of genes, the production of proteins, and the formation of new synaptic connections. Okay? So the first two portions are important. Activation of genes leading to protein synthesis. So calcium comes in and activates those genes back in the DNA, turns it and starts to create the production of proteins. Those proteins start to become folded. They go from primary structure, which you already did the labs on, primary structure, secondary structure, tertiary structures. They get put together into proteins that become, in this case, channels with receptors. Now these receptors are created, they're embedded on a vesicle which gets sent exocytosed, or rather not exocytosed, we become part of the dendritic structures of the postsynaptic neuron. So these dendrites now have additional receptors. So think about this. If I have a presynaptic neuron and a postsynaptic neuron, what are the ways to enhance that connection? Because typically that's what you would see. When repeating something over and over again in your head, what you're doing is you're constantly getting that firing of that action potential from your presynaptic neuron to the postsynaptic neurons in your brain. And if you're constantly firing it, well, the way of long-term potentiation is to increase the connection, increase the communication between the two. So what ways can we increase the rate of communication? Well, number one, you would think, yeah, produce more neurotransmitters. Okay. Do we really see that? Yeah, we see a little bit of it. Where do we can enhance the number of receptors? Yeah, we actually do. And this is the process that takes place. Build more receptors. If I start building more receptors that allow sodium in, that should excite the postsynaptic neuron. And if I start exciting it and start bringing it closer to threshold, that means it's easier to fire. Therefore, you need less neurotransmitter to cause graded potentials to create an action potential in the postsynaptic neuron. So you're enhancing that communication, and that's exactly what's happening in this case. Another thing that they find is also retrograde messengers, and I know it's a little controversial, but there's a lot more that's going into retrograde messaging. We've always known neurotransmitter leaves the presynaptic and attaches to the receptors on the postsynaptic neuron, but there's actually some signaling that comes back to enhance the communication back at the presynaptic neuron. That I won't really get into, but there's a lot that's involved in this process. Much further from what was thought originally back in 1894, where we had some earlier neurophysicists that said, yeah, we build new neurons. That's not really the case. Same neurons, just better connections between the two. Okay? And that should make sense. If I can secrete less neurotransmitter and make that postsynaptic neuron more likely to receive the signal and more likely to fire, that means there's less energy wasted. So that's our long-term potentiation. Here you can see there is some looking into neural stem cells, and a lot of key that's looking into the functionality of stem cells. And again, if you remember from our tissue lecture, stem cells are undifferentiated cells, meaning they don't have any specialization. These are the same type of cells that they're able to get from a fetus, the same type of ones that they're able to get from an umbilical cord. These are the cells that don't turn into anything quite yet. But we do have some stem cells that, even though they're starting to become neurons, you can see that these types of cells will grow and to become neurons or neuroglia. So there's some interest in the study of what's called neurogenesis, where we can create new cells. And typically what you would see and this has implications in aging, is that the hippocampus has been proven to show that they have stem cells in it. So maybe there is some mechanism to long-term memory with the growth of new neurons. And this is something that you would typically see, especially with aging, is that you start to lose some of these neurons. The connections that happen to the neurons start to decrease, and typically why people, as they age, start to become more forgetful and have a harder time learning, is that due to the connections on there. And that's really it for our neurons. Last 8 slides I have on the spine. Okay? And I don't want to get too much into it because we're going to get into, this is going to form the foundation of what we're going to be talking about in the peripheral nervous system. Everything we've been talking about so far has been the brain. And now what we're going to do is we're going to work our way into the spine. So the question, of course, ultimately, given that our test is on Tuesday, yes, this stuff is going to be on the test. It's going to add all the way to the very end of this lecture. So nothing involved in the peripheral nervous system. I know if you go to Google Drive, you'll see the peripheral nervous system lecture. Disregard that for now. Don't look at that. It's all up to the central nervous system. So I'm going to kind of speed through. Again, anatomical orientation, meninges. If you were to take a look at the meninges, we've got the same layers: dura mater, arachnoid mater, pia mater. I'm not going to get that much more into it because it's not that important. Of course the spine is important, but one of the things you're going to see is what's interesting about the spaces. So the spaces underneath on the dura mater, we have one layer in the dura mater, here, which you can see on the outside, and right underneath in between the dura mater and the and the arachnoid mater is what's called the dural space. I've got a slide that'll talk about that in just a little bit. So key difference: one layer in the dura mater as opposed to the two that you saw in the brain. Another thing we also note is the gray matter. Gray matter consists of what again? What part of the neurons?
Axons.
Cell bodies.
Cell body.
Right? Cell bodies.
Cell body.
The DNA.
And the
Right?
intra.
Dark nucleated tissue. That the neurons are located. The gray matter is on the inside, while the white matter, again, what's the white matter from?
Axons.
Myelinated axons are on the outside. So typically what you would want to see, you would see the neurons, the cell bodies located out here, and the axons projecting out, which we'll talk about on another slide. The spinal cord. So here you can see the different segments of the spinal cord, C1 all the way down to S5. Great, you've seen that already in anatomy, so I won't really go into it. Again, looming question, do you need to know the cranial nerves and spinal nerves? No, I'm not going to have you guys remember that. Don't worry about that. But what I do want to show you are the differences in the spinal cord segments. So what I want to point out is here you can see on the table to the right, here we're looking at spinal cord slices, transverse slices, and what we're going to be able to do is comparing the amount of white matter in respect to the gray matter. And what you'll notice at the very top on our cervical slices is that comparatively speaking, we have a lot more white matter than we have gray matter, right? Now take a look at the sacral slices where it's mostly all gray matter and far less white matter. And that really should make sense when you look at the spinal cord as a whole. Now looking at the spinal cord, if you were to take a look at the sacral regions, you know that they're all pretty much just cell bodies, right? And then the axons that travel out, whether they're axons that are coming in from sensory or axons traveling out from that area. Now, where would you expect to see the most axons? Axons that will travel from all these lower regions that will travel up towards the central nervous system, specifically up to the brain. Okay, so over here we see a lot more axons are projecting from lower areas of the body, making its way up. And now going back to it, here you'll see less axons traveling down more towards the tail region, and more of the axons traveling from the higher regions. So expect that when you're looking at the cervical regions, they're going to have a lot more tracks, a lot more axons traveling through. So typically, like I said, these areas will have axons that are myelinated, hence why there's white matter on the outside.
So the reason there is more white matter, or is it gray?
More white matter on the cervical
Because
region.
that's a longer distance to travel to the brain?
Well, not necessarily a longer distance, but you've got a lot of axons. You've got a lot of neurons that are located here that are sending information down those axons all the way up. So at this slice, you've got a lot of information, a lot of axons flowing through. The more axons, the more white matter. Okay. Down over here, you'll see less white matter because there's a lot less traveling through this way. So if you were to look at like the axons projecting from the hands, they wouldn't project all the way back down over here to come back this way. They really make their way over here. So everything that's innervated below that particular spinal cord segment is what it's responsible, typically where you would see the axons flowing through. The thing here talks about specifically what's called a lumbar puncture, and this is our way of getting into the epidural space, the space in between the dura mater and the arachnoid space. And what they would do here is what's called a spinal tap. This is our way of dipping into the cerebral spinal fluid, and they'll make their way in between L4 and L5. They'll stick this huge needle. They'll go in, and that's exactly what the needle looks like. It's a huge needle. And they'll come in and they'll inject right into the area, and obviously they don't want to hit the spinal cord. They want to go in and just take a little sip of that cerebral spinal fluid to see if there's anything worthy of reading into. Typically, if they look at meningitis, if you do have meningitis, which is inflammation of the meninges, they want to see, has it infiltrated the cerebral spinal fluid? Is it going to infiltrate areas of the brain? And this is their way of being able to detect that. Also, for any of you that have ever given birth, you would have had an epidural. And an epidural is an injection. So what they're doing is they're injecting an anesthetic into that area to basically block off any pain signals into that area. Pretty much block off any nerve communication whatsoever in that area. Yes?
Is there any relation to seizures? Because I've seen it done on a baby before. Who was seizing?
Was a seizure as a result of the spinal tap?
No, they couldn't get a line going, so they spinal tapped the baby.
They might have had their reasons. I don't know what would have caused the seizures, whether it was a blockage in the brain somewhere or viral or something like that. So it's probably one of their ways of being able to directly get into the cerebral spinal fluid. If they don't see anything happening in the brain, if they don't see anything happening in the blood, could they specifically see something that's happening in the central nervous system? So, just another measure. I'm sure the doctor had their reasons. That's why they make the big ones. The next thing you're going to see about the anatomy, this is going to talk about the pathway when we look at a spinal cord segment. How do we get information from the nerves located on your skin, making its way into the spinal cord and making its way back out? So we're going to be talking about afferent information going in. We're going to talk about the integrative areas that's taking place in the central nervous system and the efferent pathways out. And this slide really talks about this orientation. Like I said, gray matter is on the inside. So what we'd expect to see in gray matter are the neurons, the cell bodies. What we'd expect in the white matter is to see our axons. So here you're going to see the different areas of the spinal cord segment. I'll draw that out for you. And here I'm just drawing the gray matter. And areas that you would see that are gray, that contain gray matter, you'll see we have three different regions. The posterior gray horn. This is the area back right here. This is the posterior gray horn, also known as the dorsal gray horn. So posterior, dorsal, same thing. The anterior gray horn is this area below over here. So this would be the ventral gray horn or anterior gray horn. And then the next thing would be our lateral gray horn. So lateral gray horn are going to be these projections off the side that are fairly prominent. So typically you would see something like that on the side. So these you would see are responsible. Where you would see that on the last slide over, you'll see the sacral regions have a prominent lateral gray horn in that area. The lumbar region is a little bit more prominent, less so in our cervical regions. Now, what is the purpose of these gray horns? What you'll notice about, like you saw specialization in the brain, you're going to see again specialization of the neurons that are in that spinal cord segment. So what you would see in the anterior gray horn, in these areas again, our dorsal gray horn, these areas right here that contain the cell bodies are going to be inner neurons, and we'll talk about that in just a second. So ventral gray horn is going to contain areas right here, our ventral gray horn, are going to contain the somatic motor nuclei. And what do I mean by that? These are the cell bodies that will come in and innervate areas of the body. So if you're looking at one for the biceps, yes, it would be located in that area. If you're looking for another area in the spinal cord segment for hamstrings, sure, all located right here. Somatic motor neurons. Okay. They're located in the cell bodies over here, and they project out of the ventral root, or the anterior root. That's located right up here. Where is that? Here you can see the projections traveling out. lateral great horn contains, the areas over here contain autonomic motor nuclei. So these are things that are coming in, that are efferent pathways to, these are comparing somatic and autonomic. Autonomic would be things like smooth muscle, things like the muscles involved in the stomach, the muscles involved in the small intestine that allow rhythmic motions to move food through. Things such as the heart, our diaphragm, all of those are going to be autonomic. So going back to the first example from our lecture, we talked about the autonomic reactions of that guy almost getting hit by the car. Things that he did not have control over, his heart rate, his breathing rate. Now, the next thing you're going to see is the gray commissure. Gray commissure is the pathway. Just like you saw the corpus callosum in the brain that connected the left and right hemisphere, the gray commissure is this area that connects the left and right side. And typically what we would see the gray commissure is crossing. And we'll be able to see that on the next slide over. The crossing of axons. So tie this back into what we learned about the medulla oblongata. We mentioned that that was a decussation of the pyramids. We saw crossing that takes place in the medulla oblongata. If it doesn't cross through the medulla oblongata, which it crosses through the gray commissure. Okay? So about most of things that will cross, the things that are somatic, will cross through this area and travel back. And I'll show you that on the next slide over. The central canal isn't all that important. That's where we have cerebral spinal fluid also flowing through. White matter on the outside. So white matter on the outside, typically you would see these long axon tracks traveling up and down the spine, which I'll show you in the next slide over. And when you look at these tracks, again, bundles of axons, you have a posterior column, posterior white column, anterior white column, and lateral white column. These are, again, bundles traveling up and down the spine. The pathways, typical pathways that I mentioned about the sensory pathways, and we'll be able to see that on the next slide over. Basically, the pathway that it follows, and I'll show a little bit on this slide, and I'll really drag it out on the next slide. Sensory information flows in. You notice our sensory neurons. Again, we'll talk about more about our sensory neurons. We have our unipolar cell bodies located in what's called the dorsal root ganglion, DRG. It's this bulbous area on that dorsal root that contains our cell bodies. They'll travel through, and those axon terminals will lead to the inner neurons. Inner neurons that are going to be in between the sensory, in between the motor. So this is sensory, this is motor, ventral gray horn for somatic motor, or lateral gray horn for autonomic motor pathways. We had sensory, we had motor. What was in between? Right. Inner neurons, integrative system, right? Yeah. So if we had sensory, we had motor. Right in between is the integrative. So inner neurons are located in between. And that's exactly where the cell bodies of the inner neurons are located, up on the posterior gray horn. So we're going to learn more about these. We'll learn more about those, especially in our peripheral nervous system lecture. For now, I just want you to be familiar with the direction of travel. So sensory information travels in, inner neurons, motor travels back out. And if you were to look at the direction of travel of sensory of information, afferent information goes in through the dorsal root. Efferent information travels out the ventral root. And they're going to get consolidated into this spinal nerve or cranial nerve. Those same nerves that you see on the side of the skeleton, right, that I'm pointing at. So these pathways, and if you go into the book, you read the book is going to talk about all different types of pathways. And lots and lots and lots of different pathways. Do you need to know them? I'm not going to say no. Okay? I want you guys to be able to get the gist of it. So with this, I'll talk about the ascending and descending spinal tracts, but I will just give you just the main idea behind it. Now the pathways are basically the same pathway that I was illustrating earlier. We know sensory information from receptors. And here what we're looking at are skin receptors. So give somebody a pinprick in this area, and we have receptors. Receptors that we'll talk about that are mechanically gated. They have mechanically gated receptors located over here that will send information. That's sensory neurons, where the cell bodies are located in the dorsal root ganglion, and send that information back out into the posterior gray horn. Interneurons will send that information back out over to the somatic motor neurons located on the lateral gray. On the ventral gray horn. Any information that goes over to the skeletal muscle originates on that somatic motor neuron. Any information that travels to any of the autonomic pathways will originate in the lateral gray horn. So here you can use this, and I highly suggest blowing this up, getting it up on Google Drive, get the full color slides, and viewing this in detail. Some of the things, and again, kind of jumping a little bit ahead of what we're going to be talking about. Notice the somatic motor neuron has one pathway. It starts from the spinal cord, travels all the way to the target muscle. So severing any of the pathways of the spinal cord in any of those areas would now prohibit information traveling down to the skeletal muscle itself, leading to some form of paralysis. Autonomic pathways follow, motor pathways have one presynaptic neuron and another postsynaptic neuron. And we'll talk about what this area is, where these synapses are typically found. So there's a 2-neuron pathway for autonomic pathways, a single neuron pathway to our skeletal muscle. So sensory pathways that travel in, interneuron that comes in and bridges that information between the two, and motor neurons. What you'll also notice is skin receptors will send also this information over to the somatosensory cortex in the brain. Okay, so that would be, again, the most frontal part of the parietal lobe. That's somatosensory. If you remember that sensory homunculus, where you have the hand, you got the lips, all up on that side. Those areas of the receptors need to send that information back up to the brain, to that to the somatosensory cortex. So typically you would see axons that project, some that project on the same side, which we'll learn later on the term is ipsilateral. And then you have other ones that will cross through that gray commissure, which are going to be contralateral. Remember where it's crossing. It's crossing through that particular segment slice. This one that's going to make its way over is going to travel on the same side up the posterior white column and cross over at the medulla oblongata. Any way you look at it, things have to cross to the other side, whether it happens on the spinal cord segment or up on the medulla oblongata. So the ascending spinal tracts. Think about something that's ascending spinal tract. Why would it be traveling up? Typically, this is information coming in from sensory. This is stuff going into the brain. Conveying sensory information from any receptors, any of the somatic receptors that will be involved in proprioceptor. These are, again, motion, movement, and positioning. And that will travel up to the cerebral cortex. And here what we're looking at, that's comparing and contrasting left side versus the right side. The left side, here you can see sensory information traveling up the same side and crossing through the medulla oblongata that you can see here on this particular slice. It crosses over, and now anything that was on the right side of the body crosses through the decussation of the pyramids and crosses over. Notice where it goes to. It goes over to the thalamus. So remember, the thalamus is the sensory relay area. It sends that information out over to the somatosensory cortex. So if I were to be pricked on the thumb, that's going to go up, travel through, cross over the medulla oblongata, and cross over to that respective area on the somatosensory cortex. Other things might not, depending on which receptors we're looking at, things like temperature, and again, you don't really need to remember this, but or pain, for instance, will cross through the gray commissure. Most of them, the real heart of it, most of them will cross through. the spinal cord segment back up to and make its way to the brain. So ascending, sensory information, sending up. Now descending starts in the brain and makes its way down to that spinal cord, hence why it's descending tracks. So again, axons that travel down. If you were to take a look at where they originate in the motor cortex, the primary motor area. And those travel, again, our motor homunculus that I pointed out earlier, will travel down and either cross through the medulla oblongata or cross over to the great commissure of that particular spinal cord segment. Now, here you can see some of the different terms that you're going to be able to see on here. You're going to see corticospinal, you're going to see reticulospinal, and all of these are going to be involved in motion because these are descending tracks that travel to the spinal cord segments. And here you can see that they travel from, we have a lot of pyramidal cells. These were the cells that have heavy dendritic spines, lots of dendrites and a cell body, and that project all the way down. That travels from the cerebral cortex. If you were to take a look at the name, corticospinal. And anytime you ever look at the names of things, corticospinal, we start from the cortico, which is the cortex, down to the spine. So if I were to provide the name corticospinal, you should know that this is descending. And it's going to contain motor information. Things that travel up to the brain is ascending information. And looking at some of these, if you were to take a look at spinocerebellar, for instance, we're going from the spine to the cerebellum. So spinocerebellar tracts have information that's traveling from the spine up to the cerebellum. And what type of information is that conveying? Is it sensory or motor? Sensory. So things that travel to the cerebellum are going to be involved in sending sensory information to the cerebellum. And that's telling the cerebellum, these are the actual movements taking place. This is the sensory information that's carrying up from the legs, from the feet, from every muscle that's located, telling us what we're actually doing. I'm actually tripping. But the intended movement of the cerebellum is saying, no, you're actually supposed to be walking. So it'll actually correct it. And it'll send information back down the cerebellar spinal tract. Cerebellum, down back to the spine. That's motor information coming in, telling the leg how to correct itself, to resume walking. So just by looking at the name, you should be able to tell whether it's ascending or descending. So if I were to give you the name of a tract, you should really know, is it spinal or is it sensory or is it motor pathways? Here you see the corticospinal tract is really involved in fine motor movements, things that are very specific types of movements. Reticulospinal is really gross motor movement. These are big muscle motions that are taking place, things like posture, things that that are involved in the general motion itself. This is the refinement that takes place are the corticospinal tracts. The last thing on here, cranial and spinal nerves. You don't need to know the number of pairs. I'm never going to ask you the number of pairs of cranial nerves, where they originate. I'm not going to ask you the names of them. I will be talking about some of them. I'll talk about the vagus nerve quite a bit as we get into our cardiovascular and respiratory tracts, respiratory lectures. Spinal nerves, again, we'll talk more about that in our peripheral nervous system lecture. We'll learn more about the spinal nerves and the roots that come off of it. So be familiar with dorsal root, sensory fibers, ventral root, motor fibers. And that really sums up everything we need to know about the spine. Again, I didn't really allocate too much on the spine, more so on the brain structures themselves, but we will lead into the peripheral nervous system lecture.